So with that, we will be completing the invited lectures, and followed by that, we have the flash presentations. A bit more than yesterday, we have 15. So we will be taking up that in two parts. First 7 or 8, then there will be a question session, and then again you will have remaining people. I hope we'll be in time. Please, everyone, please do participate in this. And yeah, first speaker this morning is my own colleague, where we shared close to 3 decades of the wing and the department and the colleagueship. And Professor Gautam Kumar Lahiri, he joined just about 4 5 years after I have joined. So therefore, you can understand that we have a long association with us. And Professor Lahiri has joined IIT Bombay, I think, around 1993, after having completed his PhD from Indian Association for Cultivation of Science, followed by a postdoctoral stint in US, and then joined us and has been there. And he is a chair professor for the past 3 terms and at IIT Bombay, and he is a JC Bose National Fellow, and he is a fellow of all the three academies, and he is a silver medalist of CRSI, Chemical Research Society of India, a prestigious recognition given by the Chemical Research Society of India. And he's also been as a visiting professor. He used to spend in Germany quite often, and he used to give courses, and has a research collaboration there. And also in US, he spends time in UK as well. And he has many other awards. I hope I will probably not go into that. He is also a fellow of not only all the three academies, Indian Academy of Sciences, National Academy of Sciences of India, and Indian National Science Academy. He's also a fellow of Maharashtra Academy of Sciences as well. And he has many other awards, both from the institute side, the best teacher, and S.C. Bhattacharya Excellence Award, and quite a few, to name many more actually, is there. And he also has other awards from outside. He was a Raman Fellowship. He has got Ramana Fellowship, and he was also Edrapalli Memorial Award. He received quite a few of these. So if I have missed something, please forgive me. And he is one of the well accomplished inorganic chemist in India. And I request him without further delay to go ahead with his lecture for today. Uh, Dr. Lahiri.
Well, thank you, Professor Rao, for your kind introduction. So let me begin with by thanking Professor Rao and and his team to give me an opportunity to listen many good lectures yesterday and hopefully again today, as well as to give an opportunity to talk about some of our work over here. Now, if you see the title of my talk, which essentially suggests that we are interested in understanding the functionalizations of coordinated ligand via the mediation of the redox item, the redox at the metal ligand interface. Now, if we try to understand this particular title in with a broader perspective, it gives us the sense of two items which are moving parallelly. One, of course, the fundamental questions of electronic structures of that sort of metal ligand compounds at the interface, because we are talking about the redox active or redox non-innocent ligand, and these are a very selective class of ligands. Most of the organic molecules, they do not show the non-innocence behavior, barring few. And those kind of ligands they are frontier orbitals energy which are comparable to certain transition metals. So it creates a lot of confusion about the electronic structure both at the native and also at your redox states. So as far as the fundamentals are concerned, just a second. As far fundamentals are concerned, there are two fundamental issues one need to handle while looking into the electronic structural aspects. As I said, that this kind of ligand can behave as far as the electron transfer is concerned, like a transition metal ion. That means they can also participate in electron transfer processes. So therefore, the two aspects are very widely applicable for this class of compounds. One called the resonating form, as you understand, and also the tautomeric form. But if you go into the polynuclear setup, then another complicated aspects which comes into the picture, which we have introduced sometime back around early 2000, that is called RIET process. RIET means redox induced electron transfer process. For example, if we take a compound, a binuclear compound, ligand bridge binuclear compound in isovalent state, where the ligand is bridge is in the neutral state, and one electron transfer can be conceived in a different way. For example, if it's a metal-based electron transfer, then we should expect the mixed valence situation. If it is a ligand-based electron transfer, then we expect the radical bridge isovalent situation. And same one electron transfer process can be also thought about in terms of two electron metal oxidation with the complementary one electron ligand reduction. So essentially one electron transfer, and that's called RIET process. So we have been dealing with these fundamental issues with a variety of molecular framework, as linearity is missing over there, and some of our representative your publications are shown over here. Of course, you will see 100 publications from our group in this regard, but I'm not going to elaborate this aspect right now. Now, what we are trying to today concentrating on the fact, based on that fundamental issues, where we need to figure it out the electronic structural aspects of such kind of compounds at the metal ligand interface, how it could be applied in the form of molecular functionalization. I'm talking about the organic transformation, both at the catalytic way and also the stoichiometric fashion. Now, we have been dealing with both the things, but in the today's context, we will be only concentrating on the fact of the beta dichity minute. So before going into that aspect, let me give you two examples, catalytic and stoichiometric, and how the fundamental issues of the redox comes into the picture. For example, if you look at these two catalysts, okay, they're almost identical, except in one case that acinaptine group is there, in other case phenanthrene group is there. And as a matter of fact, what happens in this case, electron transfer is much more facile and reversible. As you could see, there are 4 steps are involved. 2 steps are shown over here. On the other hand, if you look at the phenanthrene, very irreversible, electron transfer process and high potential. So if we use these two ligands separately or catalysts separately for amine oxidation, we see under a certain reaction conditions, which I am not elaborating today. So in case of bion or acinaptine derived catalyst, we see the nitrile formation, whereas in case of phenanthrene, we see the transamination. Now, question comes how to interpret this. Though there are many ways one could think about, what we feel essentially that because of the facile electron transfer of the acinaptine group over here, we can represent this compound instead of Ru2, your neutral ligand, we can think about intramolecular electron transfer over here. So it becomes Ru3 and it becomes a radical. And as a result, this metal becomes much more electrophilic compared to the other resonating form, and that allows under certain circumstances that the amine to imine, and the second attack of imine to the metal center and gives us the nitrile. But in case of phenanthrolene, as I have already explained, the poor electron transfer feature of this phenanthrolene, it did not allow this resonating form to exist. So the amine to imine, which does not find opportunity to interact one more time because of the lesser electrophilic character of the metal center, so it goes to the transamination process. Similarly, if we look at this particular simple compound, you see that this is the 2,2-pyridyl based bridging ligand, and here there are two varieties. One variety we can consider oxygen variety, as we have shown over here. Other variety we can consider the imine variety. Now, if you react with this in a very specific metal precursor, in both the cases we get the dimeric compound in a diastereomeric form. All sort of issues are there, which I am not talking about. But in the oxygen variety, what you have seen, unlike imine variety, which is absolutely stable under any circumstances, the oxygen variety undergoes CC bond cleavage via the mediation of the small molecule activation. Here it is oxygen and gives us a mononuclear picolinate derivative. Question is why it is so? Why so much of difference moving from oxygen to the imine? The answer could be derived out of the following fundamental issues, what I have already talked about. That if you think about the oxygen attack, oxygen is a triplet molecule, it needs some kind of open cell configuration. And if we consider this dimeric configuration with the tautomeric form where metal is being oxidized to Ru3 in both the cases with this reduction over here, and this configuration does exist in case of O variety if you look at the electron energy difference, whereas in case of imine derivative the huge energy gap between the singlet to triplet, so that possibility does not exist over here, and which led to the consideration of the next step, that the oxygen molecule can interact and it passes through the dioxetane type of intermediate followed by carbon-carbon bond cleavage and oxygen bond cleavage gives us the product, and the intermediate could be trapped by the mass, and then transition state theory could also interpret how this is being formed over there. So with these two examples, I have tried to highlight the fact of the metal redox or the ligand redox, how it could tune the functionalization of the coordinated ligand as well as the molecular transformation in a catalytic sense also. Now, let us come back to the today's topic, where I would like to concentrate very specifically on a particular type of ligand, what is called beta diketominate or conventionally NAC-NAC. And if you see the literature of the NAC-NAC, you will see vast literature are mostly at the main group site. The reason is that it has a C beta nucleophile site and it reacts in a very facile way with the dianophiles and gives us the cyclization products, etc. etc. You could see a lot of literature is available. And this C beta could also activate the hydrogen also. And if we start with the coordinatively unsaturated system, then one could think about the small molecular activation, etc., etc. And one more property of this beta diketominate, it exhibits hidden non-innocence feature. The hidden non-innocence feature is that if you compare beta diketominate with the alpha diamine, now alpha diamine, the advantage is that the nodal planes are on the bond. As a result, what happened, any changes redox which would directly reflected in the bond parameters or metric parameters. So structurally one could follow the redox state. On the other hand, if you look at the beta diketominate, the nodal planes are on the atom. So it does not show the diagnostic metric parameter changes on redox. So one needs to depend on variety of magnetic spectroscopic as well as the magnetic event. Now, we thought to look into the activity or the functionalization process of this beta diketominate on a selective metal platform. What you have noticed, people have worked on this ligand in a variety of ways, but they could not activate this ligand to a large extent. So one of the aspects we took it hand, that how to behave or modify the chemical non-innocence of this ligand. So we prepared one simple compound. If you look at, and then what you have noticed, that this compound undergoes spontaneous oxygenation process at the methane center, and this gets converted to the ketodiimine function with the change in the metal redox. Okay, the question is how it is happening. To understand that, we went back to the electronic structural configuration of this compound, and if you see the precursor compound is the ruthenium 3 T2G5 configuration, 1 unpaired electron. It shows the anisotropic EPR. And if we look at the anisotropic parameters, which gives the immediate impression that this is not a purely ruthenium 3 center. And then if we see the spin density configuration, we see 20% spin on the ligand side. That does mean that instead of Ru3 NAC minus configuration, one could also think about the alternate minority contribution from the ruthenium 2 NAC dot configuration, that means radical configuration. And if it is so, then the oxygenation process could also be followed spectroscopically and kinetically in a very facile way. I'm not going to talk at length, but what I'm trying to say, that this transformation could happen in, or one could think about in two different ways. One is the anionic pathway, where the singlet oxygen must react at this center to give us the peroxo intermediate, followed by hydroperoxo intermediate, and followed by the final product, okay, through electron transfer process. But the question is that singlet oxygen consideration, which is energetically a very uphill process, but it is also known the crystal field induced triplet to singlet transformation of molecular oxygen. But anyway, this is the one of the considerations one could think about. But alternatively, one could think about the interactions of the triplet oxygen at the C beta center when the radical exists. As I said, the radical configuration could also be thought about, and then it follows the superoxo intermediate, followed by hydroperoxo, and followed by the product. Now, we could manage to trap this superoxo intermediate to the EPR spectrum over here. And if you compare the EPR of the starting one, you see that G anisotropy is much larger compared to the G anisotropy in the superoxo species. And also, if you see the spin density, spin is on the superoxo side. So we are convinced now that triplet oxygen reacts with the radical state of the ligand, and which passes to the superoxo intermediate and gives it work. And if it is the fact, then the question of the alternate anionic pathway, that means the singlet oxygen gets transformed, triplet to gets transformed to the singlet and reacts with the closed cell molecule, is it absolutely nonsense? No, it is not nonsense, because of the second example, if you see, we have taken this particular compound where there is no question of intramolecular electron transfer. So it exists in the Ru2 ligand minus state. And if it is so, we have noticed that it also undergoes oxygenation at this point, but but the question is the rate of oxygenation is much slower. And in the previous case, it is in the tune of minute, but it is in the tune of day, I should say. Okay, so we believe in what is said in the previous slide, that the triplet gets transformed to the singlet via the crystal field impact, and that reacts and passes to the peroxo, followed by hydroperoxo, etc., etc. Now, the another extreme, what I am talking about, if you take this particular compound where bipyridine is being replaced by the azopyridine, which is the strongest possible pi acceptor ligand, and there is no question of any radical formation. Okay, and there, even after a week or a month, no oxygenation process takes place. And if you look at the energy difference between the bipyridine compound and the azopyridine compound, the HOMO is being heavily stabilized over here. Then HOMO to singlet oxygen, if you see the difference, which is much higher compared to the bipyridine case, and that's why there is no oxygenation takes place. Now, if our prediction is correct that the oxygenation of beta diketominate or NAC is a function of extent of the radical imposition at the C beta site. In that context, we thought to modify the NAC ligand. So we brought into a new NAC ligand what is called the bis heterocycle methanite, this particular form. And there we have considered two varieties. One variety where there is a bridgehead nitrogen with the anticipation that this would pump in more electron density at the C beta site compared to the other variety. Okay. And then we prepared three compounds. This is the earlier NAC compound, followed by BHM compound with the bridgehead nitrogen, followed by without bridgehead nitrogen. And if we follow kinetically, all the compounds could be isolated, structurally characterized, everything could be done. Now, if we kinetically follow, then we see that where the BHM case is the bridgehead nitrogen, the rate of oxygenation is 10 times or higher compared to the NAC, as well as also it is higher compared to the BHM without any bridgehead nitrogen. The question is how to experimentally establish it. That was the kinetic factor. Now, if we see the ESR spectral profile along with the spin density, we see in case of NAC only 20% spin resides at this point, and that has been reflected in the anisotropic EPR profile, mostly metal-based spin. On the other hand, if we see this sulfur telluride BHM based, here we see some increment at the ligand side. Instead of 20, it is 26%. So that is how it is the metal-based EPR. But in case of, if we see this one, 28%, if we see the BHM with the bridgehead nitrogen, it is almost 80%, electron density at the ligand side. And if you see the EPR has got completely changed to a radical-based EPR instead of the metal-based EPR. And that reflects the why it is so fast in case of the BHM ligand. That means our proposal or our initial argument, which fits quite well over here. Now, if it is so, then naturally the reaction follows through the superoxo intermediate, followed by the same pathway as I already talked about, and molecular oxygen involvement could be established by the leveling oxygen experiment. And also, if we see the TS diagram, that in both the BHM case, that bridgehead nitrogen derived compound, where the energy gain is always there compared to the sulfur derivative in both the TS intermediate and also the final product as expected. And then if you see the resonance Raman experiment, the superoxo could be trapped very nicely, followed by, if you see the Fukui indices parameters, it also suggests that the radical-based pathway and also the mass of the intermediate. So if we put all experimental fact and theoretical things together, which completely in agreement with our initial proposition that the radical form of the compounds which could interact with the triplet oxygen, and that leads to the oxygenation at the C beta site or methane oxygenation process. That means the chemical non-innocence of the beta diketominate is a function of its extensive radical imposition at that point. Now, of course, then we moved into that first transition elements. We have prepared or reacted the same ligand with the copper or zinc. Surprisingly, here we could see, instead of oxygenation, the CC coupling products over here. So, okay, but what does it indicate? It indicates that initially it passes to the radical as we have proposed. So there is a competitive phenomenon. The competition between the interaction of oxygen at the site and the radical recombinations. So in heavier metal case, the radical recombination does not, sustain. Rather, oxygenation process is faster compared to the radical recombination. But the first transition series, It is a reverse process. So you have a lot of things to talk about, but today I would like to avoid talking on this thing. Now, if NACNAC behaves this way, the next question comes in our mind that the oxygen variety or sulfur variety would also behave in the similar fashion. That means oxygen variety means we call it ACNAC, and sulfur variety is the SACNAC. Now, if we begin with the nitrogen variety, of course, here it is, unsymmetric, unlike the previous one, we get essentially the oxygenated product. Sulfur variety, nothing happens, rather carbon sulfur bond cleavage takes place, and it gives us the mu3s derived one product, which we have already published earlier in a different context. But oxygen variety, ACNAC, it gives the, like the sulfur variety, no activity or no functionation, plus it also gives the acetylation product at the C beta site. And this cannot be converted to this one. That means this mechanism is certainly different than the other things. We have structurally characterized all these things. Now question comes, why ACNAC and SACNAC does not behave like your NACNAC? The reason what you could see, the EPR followed by the spin band density, the NACNAC under the similar chemical environment, the spin at this C beta position is around 20%. Whereas in case of SACNAC, it is 17%, and in case of ACNAC, it is 11%. So it does indicate that minor change in spin density at the C beta site could lead to the remarkable variation as far as the chemical non-innocence of the beta diketaminate is concerned. Okay, and experimentally it could be verified as well. Now, how this acetylation takes place? That question comes naturally, and as I said, that the non-functionalized product cannot be converted to the acetylation product. So we believe in that excess ACNAC in the system, which forms initially on a hydrogen bond with the ruthenium center, that increases the electrophilicity at this position at the carbonyl site, and that facilitates the interaction with the C beta site of the ACNAC, and that passes through the enaminate intermediate followed by base mediated transformation. Now, what I said over here, how do you justify it? Now, if we vary the R group attached to the aryl center, you see from withdrawing to donating group, what we see, the rate, we could follow it, kinetically, the rate of this transformation enhances rather drastically while moving from aliphatic to OMe to chloride side. That means more electron pulling in this direction increases the electrophilicity at this site, as which enhances this interaction to a large extent. Okay. Well, the question again comes, the sulfur variety, that means the SACNAC, no functionation, which is understandable. Why ACNAC is not functioning? That is the question which was essentially stalking us for a long time. Then he thought that instead of one ACNAC, if we introduce the two ACNAC, then what happens? Under the same or similar molecular coordination situation, instead of one, we have used the two ACNAC. You could see two ACNAC and one ACAC. And there you can expect to have three isomeric product, and we could crystallize all the three isomers. And we have noticed that one particular isomer which could undergo the oxygenation process, not to both the ACNAC, but only selectively one ACNAC. Why it is so? Of course, the answer is not very clear to us, but what observation what we have, that is also important. You cannot ignore that observation, that the isomer where both the nitrogen centers of the ACNAC, which are trans to the electron donating ACAC, okay, that isomer can only facilitate the functionation process. It does again indicate in a reverse fashion that the electron density at the C beta site on coordination mode, that is essentially the critical function to be deactivated by the molecular oxygen, and that indeed been observed over here. Well, I think I am at the frag end. So let me put the conclusion. Conclusion is very simple, that the oxygenation profile, which has not been looked into before we started our work in this area, the oxygenation profile of beta dictyminate is a direct function of his redox non-innocence feature. So let me acknowledge the funding agencies and a large number of my students as usual. Of course, everybody is not involved or was not involved in this part of work. Okay. So thank you very much for your kind patience. Thanks a lot.
Yeah, thank you, Professor Lahiri, for sharing a very exciting work of recent origin. I could see even some of the work is still to be published. It's really great. Uh, would you take up some a query or
Sure,
two?
sure,
Is
sure.
somebody, yeah.
Any number.
Is there anybody has any question? Because we are on time, so no problem. Don't worry. Anybody has any question query?
Already one question is there in chat box.
No, no,
You,
no, no,
you
that
may,
he has withdrawn because he has already seen that they use the tertiary butoxide. He didn't know what they have used. That's all.
okay.
Maybe I can ask one query, Professor Lahiri.
Yes.
In one of the slides, you have given the vibrational spectrum at 1066 centimeter moles for superoxide species.
Yeah.
Is that 1000? That is for what? Ovo stretch?
Yeah. Superoxo, these are all, very diagnostic response for superoxo species,
Okay.
metal bound superoxo
Okay,
species.
okay. Superoxo,
Okay,
yeah, yeah.
this is a characteristics,
Okay,
region.
perfect, perfect. Yeah. Anybody else having any query?
Sir, can I?
Yeah,
Sure,
Yeah,
sure.
yeah.
please go ahead.
Thank you, Professor Lagri, for very nice presentation. I just have a question. Could you please comment on the very beginning, bis aryl aminoacinabdolin ruthenium 2 complex?
Yeah.
How stable it is? Can we isolate or can we store sometimes for further purpose or?
It's let me put it this way. This is one of the robust ligands, okay, which
Okay,
people have handled, including myself. Okay. Now, this ligand is extremely stable. Extreme, nothing is going to happen because it's, if you look at, is the fast electron transfer takes place at minus zero point 0.6 volt versus SCE. Okay. So if it is so, then it is obvious that nothing will happen unless you put it in the right condition to undergo the electron transfer.
Yeah, exactly. I think it of how it will work if you make a ruthenium 2 complex with this one for photochemical purpose or some other reaction purpose. Yeah. Okay. Thank you, sir.
Yeah.
yeah,
Yeah, Professor Lagri, I also have a comment here.
yeah,
Sure.
yeah.
Thank you for providing a very wonderful talk. This electron transfer, which you now spoke about it, I was wondering whether did you look at any standard dependent voltammogram?
What voltogram you are saying?
Scan rate dependent voltammogram, because if you got very
Yeah.
fast scan rate, the
Yeah.
rate
Everything.
constant will match and you should be able to uh, see features,
Nothing
corresponding to the rate
happens.
constant.
Nothing happens. Okay. Now
Okay.
we can scan up to 2 volt per second. Okay.
Oh, okay.
Only the shape of the voltogram as expected it gets changed. Yeah, change means it's a slight
Distorted.
modification takes place.
Yes.
Otherwise,
Uh,
this is very reversible electron transfer. So only if you change the potential, then you can see the first two electron take up by the diamine function,
okay.
followed by the naphthalene part. Okay,
Yes.
and two successive electrons. This is very difficult process, but
Okay.
we could manage to reach even these 2 points, not in this work. So you'll see the series of our papers are there where we could reach up to this point even. Okay, so
Okay. Thank you.
it depends on what kind of coordination situation you are introducing over here. So it's not the free electron is a nonsense. The free ligand is a nonsense item. It doesn't happen anything.
Yeah.
Only
Thank you.
only one has to put the metal.
Yeah. Thank you. Thank you. I was referring ultra macro electrodes with very high scan rates. But anyway, that is fine. Thank you.
Professor Vijayamon, okay,
Yes.
you are an expert. Huh?
No, that is fine.
Don't push me in that direction.
Yeah. Thank you. Thank you so much.
Yeah,
Wonderful talk.
yeah. Okay, thank you, Professor Lahiri. Thanks a lot. So let me invite the next speaker, Professor Prasad. So while uh, connect it, Professor Prasad, please connect it. I'll be introducing you in the meanwhile. Okay, so Professor Prasad is a professor at Indian Institute of Science, Bangalore, and he has been there since 2003 or so. And he is a very well-renowned organic chemist in the country, and he joined ISC Bangalore after having finished his PhD from NCL, followed by a couple of postdocs, one in Germany, one in US, etc. Kindly switch on your video, Professor Prasad.
Yeah, I
Uh,
would. Do you mind for better streaming? I would do that
okay,
without
okay,
video. Is that
okay.
okay?
No, it would be better if we have your picture while you're talking.
No
Yeah, we
problem.
want to see you, right?
I have absolutely
Okay.
no problem. But
Yeah.
I thought
Yeah.
perhaps it is for better.
Yeah, Professor Prasad is recipient of a lot of awards. The most notable ones is the Santi Swarup Bhatnagar Prize, and he is an editorial member of the Organic Letters Journal, and he is also recipient of Young Scientist Scopus Nasi Award. Of course, he's a Swarna Jayanti Fellow, and many other things to his credit. Of course, he is a Fellow of Indian Academy of Sciences as well. So, with this very brief introduction, I request Professor Prasad to deliver his lecture. Professor Prasad.
Thank you, Professor Pulara, for your kind words of introduction. It's indeed a pleasure to be part of this meeting, and I thank all the organizing committee members for providing this opportunity to interact with all of you on this platform. So, today I would be talking about the diverted total synthesis, where we wanted to synthesize one compound but then ventured into some other compounds, like we wanted to make polyols but ended up making alkaloids. So, to tell you about what exactly we were interested in is something to do with natural products, because most of you know that the natural products are important compounds in drug discovery, and uh, most of the marketed drugs what you have contain around 50% of them the origin to natural products, or basically have their um, synthetic equivalents coming from the natural products,
There is some background noise. Yeah, now it's gone. Okay, fine.
not from mine. Okay.
No, not yours.
Okay. So basically, because of the unusual bioactive profiles the natural products show, there is a greater importance for these compounds in drug discovery. Uh, you know, drug discovery. So basically, many people have attempted the total synthesis of natural products and the derivatives, and it is an active program of research in contemporary chemistry. Now, we started our work long back, almost 15 years back, with the synthesis of these kind of compounds where you have tetrols. And then at that time, we were more keen to synthesize these compounds from a chiral pool compound called tartaric acid, where you have already two hydroxy groups, the stereochemistry of which is fixed. And then what you need to do is to convert two carboxylic acid into the corresponding secondary or tertiary hydroxy groups. For that, we picked up on a intermediate called the gamma hydroxyamides. So basically, what we are trying to do is to make the amide, and trying to tie up these two hydroxy groups, and then see whether we can make this kind of a compound and then add a Grignard reagent and then reduce that to the corresponding alcohol. For that, you need to make desymmetrization of this tartaric acid amide, where you add one Grignard reagent and then reduce it to make this particular compound here. As you see, these two amides have the same reactivity towards the Grignard reagent, so it's not easy to add a Grignard reagent selectively only on one of the amides to get to the ketone and then to reduce it. This is indeed a problem which was encountered in the literature, that when you add large excess of a Grignard reagent, you get to the diketone. And now this proton being very, very acidic, it epimerizes, and then you have no chirality associated anywhere there because of the epimerization. We serendipitously found that if you add one equivalent of a Grignard reagent, we can make the corresponding ketone if you can quench the reaction in half an hour time. And then we extended this to the synthesis of a number of ketoamides, and then these ketoamides can be reduced very selectively to these hydroxyamides without any problem. Now, if you look, we have made large number of uh, hydroxyamides starting from aryl, alkyl, alkynyl, vinyl, phosphonates, and dithiene. Now you see these are the building blocks which we used in the synthesis of natural products. And the kind of natural products we have synthesized is this kind of a range where you start from a simple tetrol derivative to that of a complex natural product called palmirolide A. All these compounds are synthesized from tartaric acid using the strategy what I have mentioned, that you add one Grignard reagent, do one reduction, and then modify the second amide to the corresponding whatever functionality you require. So our research was going very nicely with the synthesis of most of these oxygenated compounds. You very rarely find a nitrogen here except for these four indole alkaloids. You realize that most of the natural products what we have synthesized is containing hydroxy groups, macrolactones, and things like that. So it was going pretty good. So at that stage, we were involved in the total synthesis of a natural product called palmirolide A. And then we realized that if we start from tartaric acid, there are some problems associated. So we depended upon a reaction which was not explored very much in literature. That is something to do with the oxidation of furan. That if you react furan with bromine and uh, try to open it up, you can get to the corresponding diketone very easily. If you look at the structure of palmolide, this is what it is. And if you dissect it a little bit, this is what you get, the C1 to C15 portion of palmolide A. And you realize now if you correlate it to the diketone, which I have shown here, you realize that if you take a furan and a furyl alcohol like that, and if you oxidize the furan, you can make this diketone, and the diketone portion comes from this side, and which can be very easily synthesized from furfuraldehyde. This we have demonstrated back in 2011, and using that strategy where we have this furan opening as the key reaction, we have synthesized several other natural products, including the ethylamide A, and then the simatopolite 10-membered, 20-membered, 22-membered macrolactone natural products. And when one of the students from SV University, Tirupati joined me, I gave him this particular problem of synthesizing the natural product called surzine B, which is a decent glucosidase inhibitor. But this is only to demonstrate the ability of the furan chemistry, where we wanted to couple this amine and this carboxylic acid to give you the corresponding isoquinoline bond, amide bond here. So I told him that it's very easy to make this because it is from furyl carbinol, which is established in the lab. But then he might have to make this isoquinoline in a different route, because this is easily made from pyroglutamic acid. And then sometimes it is necessary to make an inexpensive compound in the most expensive way. So one of the recent, trends is to do this kind of chemistry. So he came up with this retrosynthesis, that you try to do the ring closing metathesis and then do hydrogenation to get to this isoquinoline. As I told, this is something which can be made very easily from glutamic acid. A simple pictet type reaction would give you the isoquinoline. So I told him that it's not so easy to make this vinyl glycine, because if you try to do anything with this isomerizes to the corresponding alpha beta unsaturated carbonyl compound. So the best way to do this is to take methionine and then expel the sulfur which is present in methionine. But then if you're looking for a different procedure to make this compound, what we found is that a procedure which was reported by John Ellman, where you have the vinyl magnesium bromide addition onto a sulfonamide, sulfonamine can give you the vinyl sulfonamide, and one can oxidize this hydroxy to the carboxylic acid using Jones reagent. That means this is a very robust oxidation which he has done, not susceptible for any isomerization. And then we felt that if this is the case, then we should be able to make a imine like this, and the nucleophilic addition here with concomitant elimination of this should give us even this compound. That means you're trying to start making use of the sulfonamines to do the synthesis of these two building blocks. But this is something which we did not want to do, because our aim is to do the total synthesis of this natural product. Vinyl glycine is easily available from methionine. This can be made in a different way. But then we thought, why not we make this compound by the way it has been reported in the literature, just to make in large quantities. And when we realized it, we made this sulfonamine, and when we added this Grignard reagent, we could not able to separate these diastereomers, and on top of it, the oxidation proved to be very difficult. Then we realized that perhaps it is better if we put a substitution at this double bond, so that it will be much more stable than that of this vinyl glycine, easy to handle. Only to realize that it's not so easy to make the vinyl bromides like these, which should be made by the Hunslicker reaction. So we felt that perhaps it is better if we put a hydroxy group here and try to do a dehydration of this to come to this particular alkene. Now we diverted so much so from the total synthesis of this natural product to a simple synthesis of vinyl glycine, and then started doing retrosynthesis for this simple component, how we put this, how we put this, how we put this. Then we realized that this particular component can be made very easily if you add a ketone here. That means if you add acetophenone onto an amine, one should be able to make this compound. Acetophenone is very much available in any undergrad lab. Imine is so much available in any undergrad lab. Is it so much difficult to make this compound by simple addition of acetophenone? Now, if you open the textbook, you realize there are hundreds of reactions, of manic reactions, where you take acetophenone, some proline, some other organocatalyst, some other thing will give you the corresponding amino keto compound. Surprisingly, this reaction was never investigated in the literature. Simple addition of acetophenone to a imine was not reported, except that some people use some proline catalyzed ones with aniline derived imines. So we realized that this is something which has been done by Frank Davis long time back, the use of potassium inhalate of a vinrobimide. And then one single report of the addition of a acetophenone onto a benzaldehyde derived sulfonamine, only one example, but the diastereoselectivity selectivity is pretty bad the moment you have a normal ketones. So we thought we should establish this reaction, we should investigate this, what exactly happens, can we be able to make vinyl glycines like this? And we realized that this is a reaction which goes very nicely, that if you add a base and then to acetophenone, it adds very nicely to this sulfonamine to get to this product with very good diastereoselectivity selectivity. And something which is very interesting is that if you change the imine addition from the ketone to that of a silyl enol ether, you get the corresponding diastereomer of the beta amino ketone. Now you realize the chirality is at sulfur, not at the other place. When you remove this chiral auxiliary, you realize that you have both the enantiomers of the same product. That means you can be able to synthesize both enantiomers starting from the single chiral sulfonamine just by changing acetophenone base versus acetophenone enol ether. One is the Mukayama reaction, one is the Manic reaction. Of course, one can explain the stereoselectivity observed by using proposed by Davis and others. And then to justify the synthesis, we have made a generalization of this particular process by taking large number of acetophenones and the addition to a very variety of sulfonamines, in this case alkyl and substituted ones, glyoxylic acid esters, and also having some branching here so that you can functionalize them further. And the reaction with simple lithium bases, they go very good. And then you have large number of compounds which can be synthesized. And then you realize that the acetyl pyridine, acetyl thiophene, acetyl furan, all of them works pretty good. and the reaction is so nice that you can get most of the major diastereomer either separable or crystallized or even more than 99 to 1 in the selectivity. And once we have this, we realize that this amino ketone now can be reduced to the amino alcohol, which is a prominent constituent in a number of natural products. I would like to remind you that we wanted to synthesize a natural product which is something completely different. Now we entered into how we can make this 1,3 amino alcohol simple ones. So we just took this addition product which we got by the addition of silylenol ether onto the imine to get to the beta amino ketone, reduce it, and the selectivity doesn't matter here because both are separable, and you add a undecanoic acid here to get to the HPA12, both the isomers of this ceramide traffic inhibitor. And to synthesize the sedamine part, we just took this bromo compound and then reduced this with sodium borohydride to give the 1,3 amino alcohol, and then reacted with sodium hydride to cyclize this nitrogen onto the bromine to get to the piperidine, and then remove the sulfonyl group with HCl. We get to the norsedamine, and then do a reductive amination to get to the corresponding N-methyl derivative to get to the norsedamine. So this is one of the simplest procedures of this synthesis of these alkaloids. And then once we made the addition of simple acetophenone, we felt what happens if you take a vinylogous compound that is basically a double bond in between the phenyl and the ketone. Will it be able to give such kind of a compound? And if that is the case, then you realize that this nitrogen can add now in an intramolecular Michael fashion to give you a piperidine ring. And the piperidine ring is a constituent in a number of natural products having bicyclic, tricyclic systems, and basically we can expand that chemistry from simple 2,4,1-D, which is a tri-substituted piperidine, to that of strychnine and velocimidine, which is a very complex natural product. And this is basically with the idea that if you can have the addition of this lithium anion or a thiaminylate from this methyl onto this sulfonamine, this would be very interesting to see. So we again tested the methodology, and then as you see here, this alpha beta unsaturated ketone, the addition goes pretty good, and you get to the corresponding diastereomer in more than 90-10 ratio. And again, when you add the silylenol ether, you get the opposite diastereomer with more than 99 to 1 diastereomers selectivity. Basically, you again can make both the enantiomers starting from a single chiral entity. And to demonstrate the ability of this reaction to synthesize many of these compounds, you realize that many of these ketones, what you take, have substitutions, other substitutions here at the beta position, and then you can have even a carboxylate, you can have an alkyl chain, you can have a extended heteroatoms, and then you can make on the sulfonamine, you can make many other alkyl, aryl, and many other substitutions. What was not working at that time is it doesn't work onto phenyl tertiary butyl sulfonamines, and also did not work on sulfonamines which you derive from alpha beta unsaturated carbonyl compounds, for example, ecrolene, cosinaldehyde, cinnamaldehyde, etc. What is very interesting is that if you have a potassium inhalate, that addition works pretty good to give you the corresponding sulfonamides in very good diastereomers selectivity. As I've told you, these compounds are very useful in the synthesis of these indolizidine alkaloids. So I will show you how easy we can make these natural products in 4 steps. You start from this particular sulfonamide, which you got from the addition of this alpha beta unsaturated ketone. Remove the sulfonyl group using HCl and ether, treat it with DBU to neutralize it. The Michael addition takes place, displacement of the bromide takes place. You get in a single part both the diastereomers in 41 to 35% yield, and now you reduce this ketone, you get to the hydroxy, and then do Mitsunobu inversion to get the natural product. This can be done from this ketone to this hydroxy very easily if you have selectrides, but we have not used because the cost of the reagent is very high. And again, to make this simple synthesis of this T241D, you just take this compound and do the deprotection, Michael addition, and then sodium borohydride reduction gives you 241D. Now you realize that this is 4-step synthesis, no protecting group. Sulfonyl group acts as a directing group as well as a protecting group in this particular reaction. And once we had this, as I told you, we could not add onto the alpha beta unsaturated sulfonamines the Mukayama reaction we could not perform. At the same time, we were not able to add the silylenol ether derived from the methyl vinyl ketone onto the sulfonamines to get to this particular product where you don't have substitution on this double bond. So we realize that if to make these compounds, perhaps what we require is to introduce a triphenyl phosphine, a Wittig ylide here. If we can have such a compound, if you react that with any aldehyde, such as formaldehyde aqueous solution or any other aldehyde, you should be able to make these compounds with R1 here, which is something different from simple methyl groups or the phenyl groups what you have. At the same time, an unsubstituted alkene also can be made. If this is the case, the enolate that should be added to the sulfonamine will be something like this. This is a viticulate which you make from chloroacetone. And if you can make a lithium anion here and add on to the sulfonamine, you can get to this corresponding sulfonamide. These kind of reagents were not investigated with the sulfonamine chemistry. And to make the story short, we added this particular vitigilide with LDA as the base and to the sulfonamine, and we could get to this this beta sulfonamide in excellent yield. And now you react this with formaldehyde or any other aldehyde, you can make the corresponding beta substituted keto sulfonamides. Now you realize that once you have a formaldehyde, you can have unsubstituted ones, you can have acetaldehyde, propanaldehyde, or benzaldehyde, or any other aldehydes. You realize now you can have even conjugations, you can have chiral aldehydes, you can have glyoxylate. So all these things gives you the more functionality, means you can have access to many other building blocks which can be used in the synthesis of natural products of very good biological significance. To demonstrate the utility of these compounds, we have made the reaction with simple formalin, and you realize that the sulfonamines, whatever we have taken, are of different variety. You have aryl, alkyl, and branched alkyls, as well as linear alkyl chains, and then substitutions with alkyne. As I mentioned, the more you have functionalities here, the more you can modify it to the natural products. So to show that, again, we just made the synthesis of 2,4,1-D. You take the sulfonamine, add this lithium enolate, remove it, Michael addition reduction gives you this particular compound. And then the other way is that you just take onto the same sulfonamide, you add formalin. Here, instead of acetaldehyde, if you make formalin, you can get to the corresponding unsubstituted ketone. Reduce it with cerium trichloride, you can get to the anti-1,3 amino alcohol. Remove the sulfonamide, put BOC, you can make the BOC protected one, and then protect this hydroxy. From here onwards, it's a simple palladium catalyzed hydroamination followed by arylation to give the porcine, which is a reported procedure. So we have made this particular intermediate that is meant for synthesis of porcine. At the same time, if you take this sulfonamide and then reduce it, you can get to the 1,3 amino alcohol, and then do ring closing metathesis reaction, you can get to the corresponding 1,3 substituted cyclopentene. This is basically called the carbonucleoside. Is a fashion to say, I hope cyclohexane never be called as carbaglucose. This is ridiculous. I mean, this is basically a nucleoside analog of instead of oxygen, you have a carbon. So this is something which has been useful in synthesizing some of the medicinally important natural products as well as intermediates. Now, we could add these ketones, we could add this alpha beta unsaturated ketones. What happens if you add an alkynone? Can we be able to add the alkynone to the sulfonamine? Yes, it works pretty good with very good selectivity. You can make the beta sulfonamidoketones with very nice 99:1 selectivity. Again, when you add the corresponding silylenol ether, you can make the corresponding amino ketones with the other diastereomer forming, with very good yield and selectivity. That means we can again make both the enantiomers starting from the same chiral compound. This is again a very good generalized procedure. For the first time, we could able to see a substitution next to that of the ketone is very good in adding onto the sulfonamine. I must say this procedure is only the second one to be used to add on to the sulfonamines. The first one being done by Barry Trost but he has used his prolyl ligands to add on to the bark protected imines, whereas we have used the sulfine imines to make this particular component here. And of course, the moment you have a, a functional substitution here, this you can extend to many other natural product synthesis, which we are currently undertaking. So to show the ability of this particular component in the total synthesis of a polyol, we took the ketone which we get from the corresponding enolate TMS enol ether onto the sulfonamine, and then we made the corresponding TBDPS as well as the benzyl protected sulfonamine additions, and then we reduce this ketone with sodium borohydride. We get to the diastereomers, separate them, and proceed from each of them to make the phytosyngenes, which are basically the ceramide traffic inhibitors. You know that most of the syngolipids are very, very important, biologically important compounds. So you make both these natural products starting from a single reaction, simple reaction, where you take the ketone and add on to the sulfonamine. So to, to make the story short, we started to synthesize the polyol, but then ended up having, you know, examining the reactions of sulfonamines, and then what we ended up is having addition of a simple ketone, alpha beta unsaturated ketone, with tigilides, alkynones to make simple to complex natural products. So most of the work that was carried out by Dr. Amarnath Reddy, who is now graduated and is stuck up because of the corona thing. He could not go for a postdoc, so he has taken up a job in Sinjin. Dr. Obul Reddy, who came from University of Hyderabad, working for Professor Periyasamy as a postdoc in my group, who has started this work, and Sopan is carrying out this work. And I must mention that most of the work, what has been reported, is because of a generous gift of the sulfinamide from Dr. Veera Reddy, a very close friend of late Professor Sri Krishna, who has generously supported us with this sulfinamide. Otherwise, it's a bit expensive to make the sulfinamide ligands or buying that ligand. So he was very generous in gifting us the sulfinamide. So thank you very much for your kind attention, and I would be very happy to answer any questions you have. Thank you.
Yeah, thank you, Professor Prasad. Nice, excellent. You stuck to the time as well. Yeah, we can have one or two questions. Is there any query, question? Anybody? Dr. Gandipan, you want to ask some question?
No, sir.
No.
I think it's
Hello,
too
sir. This is
organic
Kiran from
chemistry,
IISER
probably.
Tirupati.
Yeah, go ahead, Kiran.
Yeah, please.
Yeah, yeah, it's wonderful talk, sir. It's great to hear you hear your talk. And I have one question regarding this switch in selectivity when you move from like an enolate to a silyl enolither.
Yeah.
Can you please comment on that? Like, what could be the reason for that reversal in selectivity there?
As I have mentioned, see, basically what happens is one goes with the chelation to the sulfonyl oxygen, where when you have the lithium anion, it goes and binds to the sulfonyl oxygen of the sulfonamine. So the preferential attack is from the side of this sulfonyl oxygen. Whereas when you take the silylenol ether, it has no capability to bind to the sulfonyl oxygen, so it goes and adds opposite to that of the sulfonyl oxygen in an open transition state.
Okay. And also
Nothing
another
else. I
thing,
mean,
yeah, okay. So another thing is like even if the sulfonamides have the tendency to undergo like some kind of isomerization, like from cis to trans. So have you
very
observed
rare,
any?
very
Very rare.
rare, because very rare. Sulfonamides is like an oxime. Oximes will be most of the time in the trans stereochemistry. So,
Okay.
like in you see in Beckmann rearrangement, the one which is trans to the hydroxy would migrate. Similar way, in sulfonamides, sulfonamines, most of the time you get preferential E sulfonamines. Very rare to get the Z sulfonamines, except in the case of ketamines. If you
Ketamine.
make if you make it from any ketone, then perhaps you might see the easy ratios very often.
Okay. Okay. Thank you, sir. So
Okay.
another question is like,
Maybe you
it's
can
not a
post the
question.
question. You can post the question, Kiran.
Okay.
Yeah, yeah. So you can send a mail or message to him, so it will be easier. Okay. Thanks a lot. So if there is no further question, let us thank Professor Prasad for an excellent lecture
Thank
that he
you.
has given. Yeah, yeah. Let me now invite another colleague of mine for almost 3 decades from IIT Bombay, has been friend, and Professor Nanda Kishore. So I think he joined about 4 or 5 years later to me, a bit earlier to Professor Lahiri, maybe about 6 months or something. And the unique feature of Professor Nand Kishore is that a biophysical chemistry wherein one handles through thermodynamic aspects. Very rarely we have people in that. Most of the times we have people in various other aspects, but not this. So Professor Nand Kishore is a well accomplished person, a researcher in the area of biophysical chemistry using the thermodynamic aspects. And he is currently a chair professor in the Department of Chemistry, IIT Bombay. And he is also a fellow of the National Academy of Sciences of India, NASI. And he is also a fellow of Maharashtra Academy of Sciences, and several other awards and rewards, which I'm not taking up at this moment. So with this very brief introduction, let me invite Dr. Nand Kishore to present his lecture. Professor Nand Kishore. Professor Nand Kishore. Yeah. Are you muted or? Professor Nand Kishore?
Second. Now, am I audible now?
No, no, no. Yeah, I think it was muted earlier for some
Yes.
reason.
Yes.
Yeah, okay. So,
So I hope that presentation is now visible.
yes, yes.
Okay. So let me first of all thank Professor C.P. Rao and IIT Tirupati for the kind invitation and giving me an opportunity to share some of our recent work in the direction of quantitative insight into protein fibrillation and partitioning in self-assembly. So what we have been interested in understanding is these biomolecular processes in a quantitative manner. So two aspects I will be discussing today. One is the protein fibrillation issue, and second is the partitioning in self-assemblies with a broad objective of understanding the drug delivery aspects or drug solubilization aspects by self-assemblies. So since I would be talking about the fibrillation of protein, it is worth mentioning that the protein folding problem, which is how a given sequence takes up a final fold, is still not known completely. From 1962, when the Nobel Prize in Chemistry was awarded in recognition of determining the structure of globular proteins, till 2020, what is the status of protein folding problem? Computational chemists claim that they can give you give you the folding code for small peptides, but for larger peptides, we do not know why that particular unique three-dimensional structure a primary sequence takes up. So the when protein is synthesized from ribosomes, it follows a unique that from the folding pathway through unfolded, partially folded structure, it takes native form, and then eventually it converts to functional oligomers or native-like aggregate. The problem is that when it takes a alternate path from unfolded form to disordered aggregate, it may aggregate into beta structured form and eventually form amyloid fibrils, which is the major reason
for the neurodegenerative diseases. And here some disorders and associated proteins are listed. I will not go through all, but today I will present work with some of the proteins, insulin and some model proteins, for example, with the serum albumin. So the main aim is to put a cross on these alternate pathways. That is, if we can push the protein back towards the folded form, when it starts unfolding and forming disordered aggregates, then we can prevent the aggregation or fibrillation of the process. When we took a look at the literature, we found that there are a lot of osmolytes. Osmolytes are the molecules which provide stabilization to the protein under stress. So we found that these type of small molecules are able to prevent the aggregation or fibrillation of the protein. Now, taking a look at the literature, so I am showing here just couple of recent reports. For example, this article just appeared in ChemCom in about the current state of the knowledge of the protein aggregation or fibrillation issue. What we know is that a qualitative information about the formation of the fibrillation and aggregation in protein. The quantitative information is largely missing. When I say quantitative information, a process can be made quantitative by understanding the thermodynamic aspect. If we can unravel the free energy aspects, if we can unravel the enthalpic and entropic aspects, we can make the process quantitative. So we know a lot in literature about how the fibrils can be formed, but we don't know much about the reverse process. That is how we can push back when the fibrils start forming and we push back towards the native form. That information is largely missing from the literature, though recently some reports have started coming up, but once again, the information remains largely qualitative in nature. For example, let me just highlight another article which just recently appeared. Here also, the mechanism of formation of protofibrils or amyloid fibrils is largely discussed and highlighted whatever is known currently, but reverse process, once again, I'm highlighting that the reverse process, that is up to what stage the inhibitors or potential inhibitors can reverse the process is not known. So what we thought is that can we use the biophysical methods in understanding that what type of molecules or what type of functionality is important in reversing this process. Thioflavin T is an important molecule when it comes to understanding the fibrillation or aggregation of the protein. Its fluorescence emission intensity increases manifold when the protein starts aggregating in the fibrillar form. So as a function of time, one can generate the fluorescence kinetic profile as we see in the bottom right-hand side figure, where you can divide this into three parts. First is the nucleation part, second is the elongation part, and third is the mature stage fibril formation. But this kind of kinetic profile is not observed in all the proteins. For example, it is observed in proteins like insulins or some other intrinsically disordered proteins, but the model proteins like serum albumin do not show this nucleation step. But nevertheless, if we see in the right-hand side figure, the upper curve which shows a sigmoidal kind of behavior It shows the formation of the fibrils, but the purpose is to prevent the fibril formation by the use of inhibitors. We largely use isothermal titration calorimetry in understanding the mechanistic aspects of these biomolecular interactions. Standard molar Gibbs free energy change, standard molar enthalpy change, and entropy change. These are great indicators of the nature of intermolecular interaction. So our effort is to unravel the nature of intermolecular interactions in terms of changes in enthalpy or changes in entropy. So just as an example, for example, in HIV-1 protease inhibitors, over a period of, let us say, about 10 years or 12 years, what is it emerged is that from the first-in-class to the best-in-class kind of drug, the polarity of exothermicity of interaction turned out to be the important factor. Same thing was observed in the statins. So what I want to highlight here is that the thermodynamic signatures are very useful in devising guidelines for rational drug design. So when we talk about the interaction of a given inhibitor or a ligand with the protein, the various factors that need to be considered are desolvation, hydrogen bond, van der Waals interactions, and conformations. Now let me present some of our recent work in this direction. This slide shows the fibril formation in insulin. What we see here in the right-hand side upper diagram is distinctly three parts: the nucleation part, elongation part, and the mature stage formation of fibrils. And this TEM image clearly shows very nicely formed fibrils. And what we observed that when we use the osmolytes such as betaine, citrulline, proline, and sorbitol, we can see here in the bottom D panel that the THT emission intensity goes down when you increase the amount of osmolyte in the protein. And this shows that these osmolytes are able to prevent the fibrillation in proteins, that we can compare the microscopic images. But our main purpose was to understand the enthalpy of interaction of these potential inhibitors with the protein at different stages of the fibrillation process. The strategy that we adopted was to take out the sample at nucleation stage, elongation stage, and fibrillation stage, and then interact these samples with the inhibitors, monitor the thermodynamic signatures, and then try to find out what type of interactions are taking place. Now, as we can see here in this lower right panel, the typical interaction profile, although I have not shown here the raw ITC profile, it was not of a binding type of profile, because if there is a sizable binding, moderate to strong binding, one will get a sigmoidal kind of behavior. But since the protein at different stages of fibrillation will not have specific binding sites for these inhibitors, therefore it is not going to show a sigmoidal kind of behavior. What you will get is a linear kind of behavior. So the challenge lies in extracting or unraveling the nature of intermolecular interactions from this kind of data. So one way is that we find the infinite interaction parameters, or infinite, the limiting parameters of enthalpies of interaction. So what we do is we try to find out the enthalpies of interaction of the inhibitors with the native state, elongation state, fibril state, and then compare this endothermic or exothermic nature of interaction. We understand that if the interaction is endothermic, then what type of interactions will dominate? Endothermic interactions means that the hydrophobic effect is predominant, or the desolvation of the molecule which is interacting may be predominant. Exothermic interactions highlight the role of the polar interactions. So we could propose even a mechanism of the prevention of fibrillation of insulin by using this combined calorimetric, spectroscopic, and microscopic approach, and show that the osmolytes are able to inhibit the formation of fibrils, but sometimes these do form small amorphous aggregates. And of course, this step is also not the wanted step. Can we use differential scanning calorimetry in understanding this kind of phenomena? Well, if we look at the left-hand side panel on this figure, where A, B, C, D, E, these are all, A is just for the buffer versus buffer baseline, and B is the native protein. And as the fibrillation extends, we see that the extent of endotherm decreases. So quantitatively, we can tell that how much the protein retains its structure in terms of enthalpy. And when we talk about absolute heat capacity, we see that the native state, elongation states, and saturation states heat capacities are different. And this increase in the order NES, obviously because the extent of structure, extent of the aggregation or fibrillation structure increases when it moves from native to elongation to saturation state. So we know that how do the amyloid fibrils form? There is a lot of literature existing, and mechanistic models are available. How do the fibrils form? For bovine serum albumin, let me show now this some data on bovine serum albumin model protein. What we have is, here we don't see the nucleation stage. We right away see the downhill kind of polymerization process. So here also what we did was that we took the aliquots out at different time points and then interacted with the potential inhibitors. Of course, side by side, it is important to characterize the different stages of fibrils by microscopic and dynamic light scattering measurements and see the size of these aggregates or fibrils formed. So let me not go into that. So next we wanted to check that what type of molecules are able to provide a better inhibition efficiency. So first we tried with the polar drugs, and we wanted to check whether the polar drugs can inhibit the fibrils, whether intramolecular hydrogen bond is formed between drug and beta strand, and what is the role of intermolecular interaction. So let me show some data with two drugs here. I will not be able to show with the other drugs because of shortage of time. So this is a hydroxyurea, which is used in anti-cancer, which is known as an anti-cancer drug, and it has several hydrogen bonding donor and acceptor groups. So this molecule can participate in hydrogen bonding. For example, if these are the sheets which are coming together to form the fibrillar structure, can this type of molecule sandwich in between the associating fibrils or not? We wanted to check by the use of isothermal titration calorimetry in addition to the other required biophysical methods. So what we do here is we compare the enthalpies of interaction of this molecule with the protein at different stages of the fibrillation process, and then we try to correlate with the functionality on the molecule. So I will rather show, okay, the DSC also is important. I will not go too much into details of DSC, but let me show some data with another, again, anti-cancer molecule, which has been used in the treatment of breast cancer, is 5-fluorouracil. Here, this molecule has also the hydrogen bonding groups, which can participate in hydrogen bonding. So in addition to the quantitative estimation of the fibrillation process based upon the THT emission, uh, our main aim was to understand the role of functionality on these molecules and its extent in reduction of the fibrillation process. Well, from the studies, one can also find out that how much the reduction in the fibrillation is possible by different drugs, but the extent of reduction in fibrillation may also be accompanied by the formation of amorphous aggregates, which is not a desired process. For example, in these two cases, hydroxyurea versus 5-fluorouracil, we can see here that how much is the extent in reduction possible in the early stage or the later stage, but the results also showed that in hydroxyurea there were formation of the amorphous aggregate. So now this data is interesting. In 5-fluorouracil, if we just check the enthalpies of interaction of this molecule with the protein at different stages of the fibrillation process, we see here the exothermicity decreases when you move from native to elongation or protofibrils and then to mature stage fibrils. We can see here also it is more towards little endothermic when the mature stage fibrils are formed. So this gives an indication of the changes in the polar interaction between this molecule and the associating protein molecules. And of course, along with we also took support from the F19 NMR spectroscopy in understanding these intermolecular interactions or the participation of this fluorine group in hydrogen bonding in the associating molecules. Another important technique which has not been widely used in the understanding of this fibrillation or prevention of fibrillation process is pressure perturbation calorimetry. Many groups, they use the differential scanning calorimetry in understanding the folding unfolding process, but there is another feature which is present in differential scanning calorimetry is pressure perturbation calorimetry. What you do is that you increase the pressure or you fluctuate the pressure and then you monitor your signals. Well, the physical chemists know that dG is equal to VdP minus SdT. That connects the Gibbs energy with volume and with entropy also. So PPC makes use of that and connects the Gibbs energy changes with the volume. So you can actually calculate the volume changes, you can calculate the expensivity, and then all these things can be connected in terms of exposure of hydrophobic residues or exposure of the polarity during these interaction processes. For the lack of time, I will not go into this. So what we could, this work has just appeared in just accepted manuscripts in JPCB. What we tried to show here is that the 5-fluorouracil molecules are able to sandwich between the associating this beta sheet strands and able to prevent the fibrillation process to appreciable extent even when these protofibrils have started form. But once the mature state fibrils are formed, then this molecule is not able to enter in and separate the associating strands. So obviously, this all information is based on the enthalpies of interaction and with support from the other biophysical methods. So similarly, we were able to show that even the molecules, surfactant type of molecules, are able to disintegrate fibrils. And of course, disintegration may not give you a clear picture, it may give amorphous structure, because these type of molecules which inhibit the fibrils are not able to push back in towards the native state protein. So I will also highlight another aspect of our study where we focus on the encapsulation of the drug molecules into the vesicles, micelles, or self-assemblies, and the consequent release and binding at the target protein. So very briefly, I will present here. What we try to do is, for example, if I take a very simple self-assembly system here, the micelle, and the micelles have a hydrophobic core. Micelles can be used for the solubilization of the hydrophobic drugs. We know that the majority of the new molecular entities, they fall into the category of hydrophobic moieties, and therefore their delivery requires the solubilization and then appropriate delivery or efficient, effective delivery at the target site. So we started by choosing the molecules like the sodium or this is H-tag micelles. Can we understand quantitatively that when we interact the drug molecules with it, where do the drug molecules go and sit? They sit at the surface. Are they able to go inside the hydrophobic core? And in order to achieve or in order to tune this partitioning, what type of functionality can be modified? So this can be addressed in terms of the physicochemical properties. So if we try to understand some physical chemistry of this, for example, this first equation XS forming SX, this is a simple micellization process. And then Y moles of drug can partition in this micelle, form a drug partitioned micelled complex. And then we can actually try to find, where we can actually calculate how many drug molecules have partitioned into the self-assembly. And one can talk about, where these drug molecules are actually going and partition. So the extent of partitioning will also decide the extent of delivery. And this extent of partitioning or connection with the enthalpy, although I have not shown here enthalpies of partitioning over here, the enthalpies of partitioning actually tell us about the nature of interaction. For example, if it is partitioning deep inside the core, we will get the enthalpies towards the positive side. And these values of K, the partitioning constants, will tell us the extent of partitioning of these molecules into the core. So one can, the eventual purpose of this is to make structured property energetics correlations. Because if we can identify that what kind of properties or what kind of functionality can lead to better partitioning, both in the drug and the drug partitioning system. Now I have showed only the micelles. We have also done work with the neosomes, which are better drug, which are actually used in drug delivery systems, and the work can be extended to other drug delivery systems which are in use. So we tried to make a structured property correlations to understand that in this kind of system, is there any correlation between the functionality and between the values of K, H, and delta? What we observed from a certain set of data sets, that there is no direct correlation between the number of hydroxyl groups, number of NH2 groups, or number of NH groups, but a good correlation was seen with the size, the steric factor. So that means the steric factor and size becomes an important parameter over here. And of course, the other functionality will also contribute to where the drug molecule will go and partition. So based upon these thermodynamic signatures, we were able to get a mechanistic aspect of how the drug partition in self-assembly, And once it is partitioned, then you study the release, interact with the protein and see whether the released drug binds the protein at the same binding site, and whether the components of this drug delivery system, they affect the conformation of the protein. So these kind of intricate details were possible to be obtained by using the combination of calorimetry and spectroscopy. So we went on to study the interaction with the drugs which varying these macrolide based drugs, where you change the basic, the core structure, and see the how the partitioning takes place. So due to lack of time, I will not go into detail, but highlight that it is important to understand that how the components of these drug delivery system affect the target protein. That can be achieved very, very efficiently by using differential scanning calorimetry, because it will show you in terms of enthalpy of unfolding that what is the effect on its conformation or on its structure. Of course, there are other spectroscopic techniques like circular dichroism spectroscopy, which can be used in this. So we went on to study the physicochemical aspects of drug partitioning and drug release in more complicated systems for neosomes. And here we were able to even show the drug release kinetics and obtain these rate constants. That these rate constants, we actually drug partitioning is still well reported in literature, but quantitative aspects of drug release is not that much reported in literature. But this we were able to show by the use of isothermal titration calorimetry. What you see here in the bottom panel here is the drug release by ITC. So what we did was we planned the injections that, for example, separated by 2 hours or 3 hours. And then we try to analyze here the each peak in the, for example, what you have here is the power versus time. So anything which varies as a function of time, showing this kind of behavior, one can extract the kinetic parameters out of it. So it is possible to obtain the thermodynamics and kinetic information from this. For example, this is just the expanded view. So to summarize, the combination of calorimetry and spectroscopy can give us a detailed understanding of partitioning of drug in self-assemblies. Here I have shown by choosing a very simple system, that is the micelles. By doing some physical chemistry on this, we can understand the partitioning constant, we can understand the enthalpy change, entropy change, and then we can also use a combination of ITC and DSC to test whether the drug delivery system along with the drug, when it is released, does it have effect on the binding efficiency, and also whether it affects these drug delivery components, they affect the structure and conformational integrity of the protein. So in short, when we use these thermodynamic aspects and focus on the enthalpies, enthalpies carry a lot of information, because the sign of delta H, it carries information on the nature of interaction. And of course, the other parameters like binding constant, these of course can be determined by the use of the spectroscopic method. And the equilibrium constant, binding constant, enthalpies can also be determined from spectroscopic methods. But calorimetric methods give the direct information on the, or what we say is the true thermodynamic signature. Actually, I could not include that slide here. We analyzed the thermodynamic information available in ligand binding reactions by using isothermal titration calorimetry and by using spectroscopic methods. And we find a big discrepancy between the thermodynamic information available by using these two techniques in a variety of ligand binding reactions. Some cases it does match, many of the cases it does not match. So we have analyzed this, and this has just come out as an article in, again, it is in the ASAP publications in Bulletin of Chemical Society of Japan. So where we analyze these discrepancies and have suggested a protocol that how we can resolve these discrepancies between the spectroscopy and calorimetric information. So I conclude my talk by saying that the thermodynamics has a lot of potential in uh, in providing the quantitative aspects of the ligand binding reactions and biological processes uh, uh, in unraveling the intermolecular interaction and hence contributing to rational drug design. So in the end, I acknowledge the financial assistance in a big way from SERB, BRNS, IIT Bombay, and of course, I have been lucky to get excellent PhD students. Thank you very much.
Thank you, Professor Nandkishore, for an excellent work and excellent publications. Really good, but I think we are a bit too, overshot the time. So there is some question from Professor Vijay Mohanan. So if it is pressing, you go ahead. Otherwise, we will go to the next speaker. Professor Vijay Mohanan.
Yeah, it's okay. It's a very minor one. I saw
Yeah.
some nodes in the TEM. I was
Yeah.
wondering whether there is a network structure is forming or not.
Yeah.
That's okay.
So
We can
yeah,
talk. I can talk to you separately. Don't worry. You go ahead.
really, thank you, Professor Nandkishore, for a very nice lecture. In fact, that pressure perturbed calorimetry. So I did not know basically. So the new things, and we also have shared some work in this direction for metal ion-induced fibrillation and defibrillation kind of things, which we will discuss later, of course, between us. So thank you very much for a nice lecture. So I'll call on the next speaker is my senior colleague at IIT Tirupati. So this is Professor Dr. Debashish Mondal, who joined IIT Tirupati in 2017 after having finished his PhD from IACS Kolkata and then a few postdocs, followed by that one in Virginia, another in UMass, and so other in Dresden, and then joined IIT Tirupati in 2017. So he's been working in stochastic processes. Obviously, I will not understand. And he's working on physics of polymers, physical properties of active matter, principles and applications, etc. He already has been actively working and started publishing as well, and received the projects, including the Early Career Research Award from DST. And he's on the review of editor and the editorial board of biophysics frontiers in physics in this. So with this brief introduction, I request Dr. Debashish Mondal to present his work. Dr. Debashish. Dr. Debashish? Is there some problem with your...
So your audio is
Can,
We can
can
see
it?
the slides.
yes. Thank you, Professor, for your kind introduction, and thank you for providing me such wonderful opportunity to share some of our recent developments to this fantastic audience. And good morning, everybody. So today I will discuss about noise-induced symmetry breaking of self-regulators, and this will lead to an asymmetric transition towards the homochirality. If we consider the chirality of an object, then what picture come to our mind is that the chiral objects or enantiomas are nothing but non-superimposable mirror on its mirror image, either by translation or by rotation. So these non-superimposable mirror image enantiomers, actually they are chemically similar in nature. However, if one passes some plane polarized light, then the plane of polarization rotates clockwise in clockwise direction for some cases, say for example in terms of D quad, and anti-clockwise or counterclockwise direction for some other cases. So therefore, chiral objects or enantiomers, they are chemically similar, however, they are optically distinct. Now, why these chiral objects are very important in life? So, as we all know, the building blocks of DNAs and RNAs are amino acids. And almost all the amino acids that found in living organisms naturally are of L type. On the other hand, if we consider the type of sugars, we can see that almost all the naturally occurring sugars are of B type. So that means in life or in nature, there is some spontaneous or automatic symmetry breaking, which is called homochiedality in nature. And as Professor Ganesh was mentioning yesterday, that nowadays this is connected to the origin of life. This understanding this issue becomes one of the holy grails in chemistry. So as you all know, if we try to synthesize any organic molecule which is chiral in nature or some optically active crystal from some achiral substance or materials, what we find? We find a racemic mixture. So then the question arises, why organic molecules in living organisms are homochiral in nature? So to perceive this, people were trying to understand the reason, and there are some speculations. It is believed in both in chemistry and physics community that there might be associated two stages. In the first stage, there might be some initial creation of chiral bias or enantiomeric imbalance in the system. And this enantiomeric imbalance might come from different reasons. Some of the speculations are because of the rotation of Earth in its around, or sometimes people find that it might be because of some extraterrestrial reason, as meteorites that are found are having little bit more excess amount of L amino acids. Now, once we have some kind of initial imbalance or chiral bias into the system, people believe that there must be some type of autocatalytic mechanism along with some mutual destruction. With that, this chiral imbalance will grow and it will give a stable and steady homochiral state. So therefore, based on this speculation and idea, people were trying to rationalize the reason for emergence of homochirality. The first theoretical model in this regard was proposed by Frank in 1950s. And what he assumed that he took one three-state model system, and he assumed that one molecule or chiral, if it may persist in three different isomeric form, out of that one A is the achiral in nature. I'm sorry, my okay. And other two, D and L, they are chiral. And he proposed that there might be two different reaction setup. One is autocatalysis, where either L or A will produce more amount of L and A in presence of achiral species with some associated rate constant. And he introduced another reaction process called chiral inhibition, where two enantiomers will mutually destroy its optical activity to give some achiral species. Now, if we combine these two reactions and express them in terms of simple reaction kinetics, what we can have that in stationary state or steady state, the non-trivial solution, if we consider the concentration of the achiral species is very large and constant, so you may have that the concentration of B and L form will be exactly similar or identical. That means we will arrive to one racemic situation. However, if we introduce a little enantiomeric bias initially, that means the initial concentration of D and L, if they are different, what we can find that this small enantiomeric excess can be amplified with time and can give rise to the homochirality or symmetry breaking situation. So the first experimental proof of Frank's model or Frank's assumption was framed almost 4 decades later in 1995 when Soai and his group was trying to synthesize 5-pyrimidine alkanol from associated carbaldehyde in presence of diisopropyl zinc. What they have found that if they incorporate a trace amount of the one of the enantiomer of alkanol, the symmetry breaking was happening, and they were finding that the more and more amount of that isomer can be found. However, this symmetry breaking was partial in nature, not 100%. But later, in 2005, Viedma et al., they were doing some experiments with sodium chlorate crystal, and what they did, they took some sodium chlorate crystal, and there was D and L from 50-50 situation, and they add some glass bead and start the solution. Now, if they found that if we add some initial enantiomeric bias, either by in terms of D or L, so when it recrystallizes after a few hours, you can have only that fall. So from here, they also support the concept and the assumption that Frank has been proposed 40 to 50 years earlier. Now the question arises, so far we have seen that if there is some initial imbalance, we can give it externally to the system, then that might lead to the complete or partial symmetry breaking the system. People were trying to understand whether this enantiomeric excess, the form of the initial enantiomeric excess, can come or can generate it by the system inherently. And the answer is yes. So what people find that if we can create an stochastic environment where all these associated reaction process, not only just autocatalysis or chiral inhibition, what we have discussed, so they incorporated, one can incorporate another non-autocatalytic pathway of decay of the achiral species either to D or to L form, that is called achiral decay. Now, if all of these reaction processes are stochastic in nature, then actually people found that even in absence of any initial external bias in terms of enantiomeric excess, one can have the symmetry breaking situation. However, there arises some confusion. In 2015, Jaffarpur et al., they were trying to model this thing using Gillespie algorithm, and what they have found that it is the achiral decay along with the autocatalysis process which is important to have a symmetry breaking situation. On the other hand, D. Bhattacharya et al. Recently, they were trying to mimic the Frank's model without considering achiral decay with some different type, little bit different type of reaction mechanism, and they also found that just like Frank model, autocatalysis along with chiral inhibition is very important to have this symmetry breaking situation. So one of our aim of our project, how to reduce this confusion or to make the model much more generalized, one. Second is that most of the theoretical studies are talking about only symmetry breaking. We are interested to see how much symmetry breaking happens under which reaction condition, whether it is complete or partial. So in this spirit, what we did, we also considered a three-state model where any molecule may exist in, we assume that that molecule may exist in three different species. One is achiral A, and D and L are chiral species. We consider chiral inhibition and achiral decay process as Arrhenius type reaction, that is reaction over some thermal activation energy barrier. So along the reaction coordinate, definitely D and L form will have similar energy. A may or may not have similar energy, that might be less than or greater than that of the D and L form. Now, this both the chiral inhibition and achiral decay we map these two reactions in terms of Arrhenius type reaction process, that is for chiral inhibition. It is confronting this activation energy barrier to be mutually destroyed to have achiral species, or for achiral decay, A can be decayed either to L or to D along the reaction coordinate over this second activation energy barrier. However, we introduce the autocatalysis reaction process for the same in a little bit different manner. So let us explain this. So what we did, we assumed that all particles are Brownian particles or Brownian molecule in nature, and they are moving randomly in this two-dimensional space. We did it in 2 dimensions. It can be easily extended to the higher dimension as well. And what we considered is equation of motion of this particle in space is governed by this Langevin equation, which is very standard. On top of that, we add one additional degree of freedom along the reaction coordinate, which is sigma. So by this sigma, we are taking 3 states. If it is in this state, we are considering the value of sigma I is of the right particle is -1. If it is in L, then it is +1 and 0 for achiral state. So along with the spatial reaction coordinate, we along with the spatial diffusion, we incorporate another additional degree of freedom along the reaction coordinate, which is sigma I. And then to mimic the autocatalysis process, we incorporated an Voters model. So what we assume that if the molecule is in achiral state, this yellow color is in achiral state, it can interact with other molecules within some radius of interaction, and we set this radius of interaction from outside, of course, and we look at how many D particles are there inside this radius of interaction. If the D isomers are dominating over L isomers inside this radius of interaction, then this achiral species will be converted to D. Okay, so this model is inspired by Voters model, which was first introduced by Kim Flood long ago in 70s. So what he assumed, suppose some person is politically unbiased, and there are in that state or in that nation, there are 2 parties, red party and green party. Now that if that politically unbiased person is surrounded by more red party supporters, slowly that person's mind, political mind, might change to as a red party supporter or vice versa. Okay, so this model we introduced here, and as I mentioned that the other 2 reaction process, that is chiral inhibition and achiral decay, we consider those reaction as Arrhenius reaction process. So if we combine all these 3 together, we can express the time evolution of these additional degrees of freedom, that is sigma, with this relation. Now, to study the symmetry breaking situation, we introduced one order parameter. So order parameter is will give us the amount of enantiomeric excess if there any. So we define it as the summation over all the sigma i's value, that additional degrees of freedom, and we take its modulus value, and we took the average of it. Now there will be 2 limiting situations. One is that if we have a residue mixture, that is 50-50 D and L, or all particles are in achiral state, we will arrive phi t equal to 0. On the other hand, if we have 100% or complete symmetry breaking, that is 100% particles are either in D state or in L state, we will have this phi t value 1. That means that is homochiral state. So if we consider this thing and if we simulate the system, so what we did, we took a situation, we considered a situation where there exists no initial imbalance, that is A, D, and L, we take 1/3, 1/3, and 1/3, and we find that with time, this order parameter of the system is changing if we change the reaction coordinate, and we can see that based on the differences in reaction condition, we may arrive to different situations. Sometimes we may arrive to a complete symmetry breaking. This black line, you can see here the order parameter is going to one that is 100% symmetry breaking. In another reaction condition, it is symmetry breaking is there, but this is not 100% in nature. So we also define this phi s as a stationary order parameter at long time. Okay. Now the question arises that how many different type of stationary states we can have from this system, and whether is it possible to have some non-equilibrium transition from one state to another if we change the reaction condition. In this spirit, we identified 4 different population state or asymmetric state, I should say. One of that, if we consider the rate of both achiral decay or chiral inhibition is very low, and if they are equal, in this situation, whenever I am saying the rate, it is associated to the relative to the rate of autocatalysis. So we scale all our rate constant in terms of rate of autocatalysis process. Now, in this, under this situation, we can see that 100% conversion of one of the isomer, in this particular case D, and we denote it as homochiral state. Please note that this is one trajectory. So if we change the trajectory, it might be 100% L. So in this study, we cannot say whether it will be D or L. We can only say under this reaction condition, there will be 100% symmetry breaking. Now, if you incorporate a little initial imbalance, then based on the type of the imbalance, you can say that definitely under this reaction condition and with this imbalance, it will go to D or L. Another situation we have when we increase the relative rate of both chiral inhibition and chiral decay. That means we are changing this associated energy barriers of these reaction processes. Then for high rate of chiral decay and chiral inhibition, what we can have a racemic type of solution where actually 3 isomers can coexist. We denote it as 3 isomer coexistence state. Similarly, if we now leave the constant that the equality of this rate of chiral inhibition and chiral decay, and if we consider the stronger chiral decay, that means this blue line, this barrier height is less than that of this chiral decay, chiral inhibition, red barrier. So in that case, we can see a partial symmetry breaking. And here we notice that 2 enantiomers may coexist at stationary condition with some unequal distribution. Similarly, if we reverse the reaction condition, if we take a stronger chiral inhibition than chiral decay, we notice that there will be a situation where chiral component along with that any one of the chiral one can coexist. So this we call chiral coexistence. So these 4 types of asymmetric state at stationary condition we have noticed. Now we tried to scan the phase space in terms of reaction rate constant to see in which under which reaction condition we can have any one of these asymmetric state, and whether from there in which direction if we change the reaction condition we may have some transition, non-equilibrium transition of state or not. So in this spirit, we scanned the chiral decay rate constant along with the chiral inhibition rate constant, and what we have find that, so if we consider a point near P, so here both Kd and Ki very small. That means both of these energy barrier are extremely high, and these chiral decay and chiral inhibition rate is extremely low comparative to that of the autocatalysis. Under this situation, because of the Votter's model and autocatalysis reaction, you will find quickly, so all chiral molecules will be converted either to D or to L, and because of the stochasticity of the system, there will be very minor differences in concentration. It might be 51-49 or 52-48 like that. So there will be creation of an enantiomeric bias, but there are no proper or suitable reaction mechanism which can enhance this enantiomeric bias. So this is the situation we arrive, and you can near this point P you will have only 2 enantiomeric coexistence situation. Now If we consider the point Q, where we consider the rate of chiral inhibition is very high compared to that of the chiral decay, you can arrive to a situation, the energy profile will look like this, arrive to a situation that in stationary condition, you will have some residual achiral moiety along with one of this D or L. That means because of stochasticity, there will be creation of the symmetry breaking, initial imbalance. But before this converted to a 100% symmetry breaking, there will be a equilibrium, or sorry, not equilibrium, stationary situation where you can find some residual amount of achiral species. So this is state 2. And finally, if we try to move along this line, what we can find that there will be some optimal position. Let us consider this x, point x, where ki and kd are equal to each other, but it is much, much less than that of the rate of the autocatalysis process. We can have actually the complete symmetry breaking, that is homochiral state. Now, once we are in this homochiral state, so staying here, if we change the one of the reaction rate constant, let us see what happens. If we change the reaction rate constant in terms of chiral inhibition, you can see that beyond some range, if we increase either or if we decrease, we will lose the concept of complete symmetry breaking, and there will be a transition from complete symmetry breaking to partial symmetry breaking based on the reaction rate. On the other hand, if we keep the rate constant of this chiral inhibition unaltered, and if we change the achiral decay rate constant, you can see that even if we switch off the achiral decay, we can have the complete symmetry breaking state. However, if we increase the rate of achiral decay, then we can arrive to a situation where there will be a transition from complete symmetry breaking to partial symmetry breaking, and we will lose the essence of the or situation that may give rise to the homochiral state. So therefore, we can say that it is the controlled amount of chiral inhibition which is very necessary along with the autocatalysis process to have the complete symmetry breaking state. Of course, on top of that, there might exist some amount, in some extent, the achiral decay, but if the achiral decay rate is too high, it may spoil the game. Finally, what we did, we considered a packing fraction like parameter that I was talking about, the interaction radius R. So how much interacting neighbors are there inside that interaction radius, that is NR, in terms of this total number of particles, box size, as well as the chosen radius of interaction. And we try to calculate the order parameter with that NR. What we have found that if this NR is very low, then we have a racemic mixture, and if it is above some threshold, we can enter to a homochiral state. So there is a crossover from racemic to homochiral state through a partial symmetry breaking situation. These different color points here denote that the same NR value with different combination of N, R, and L. That means in this way, we can say that this need of this threshold in terms of NR is universal, and it doesn't matter if we change the radius of interaction or the concentration of the molecule or the box size. So it is overall this NR, that how much neighboring interacting particle is there inside that interaction radius, that matters to show us a partial or complete symmetry breaking state. So finally, to conclude, so what we have that, of course, we may have continuous symmetry breaking without any initial bias, and we identified that there might exist 3 other different type of states based on the different reaction condition, and system may undergo asymmetric state transition if we change the reaction condition. And if we consider the complex symmetry breaking or homochirality, it is a first disordered self-regulation or autocatalysis process along with a controlled amount of chiral inhibition. And of course, we do need a threshold number of the interacting neighbors that is necessary and sufficient to give a complex symmetry breaking situation. And of course, achiral decay may be present in the system, but that is not an crucial state to have complete. So finally, I would like to acknowledge Mr. Rafna Rafiq and my other group members. So actually, Rafna did this work as her BSMS project. She was a BSMS student from ISA Tirupati, and I was lucky to have her as my student. And in fact, she did all this work, and kind of I was just along with her, and also some of the funding agencies. Thank you.
Yeah, thank you, Dr. Debashish Mondal. So yeah, so now, is there any question to Debashish? Anybody would like to? Because we are a little bit delayed, but still, if there is any question, then we can address. Any question to Debashish? Yeah. If not, let us please join me in thanking all the 4 speakers this morning. Excellent presentations that we have on this platform today morning. So thanks to all the speakers and to all the participants for listening and interacting with it.
Thank you, sir.
Now, yeah, thank you. So now we will have the next session, which is the flash presentations. So the flash presentation, like we have, I think, about 15. So this will be handled by Dr. Arun Manna, my again senior colleague in the department. So he'll be handling this. So Dr. Arun, you please stop after 7 or 8. We will interact for 5-10 minutes. Then again, you can continue. And second thing is, if somebody is not there, please don't wait. Go to the next so that they can come back at the end. Okay. So the floor is to you, Dr. Arun Manna. Go ahead.
Thank you, Professor Rao, for the platform, and good morning to everyone. So I welcome all of you for this 4th technical session. And this is formally the last session of this 2.5 days program that we have. So we have 15 presenters for the flash presentations. And as already Professor mentioned, after 7 or 8 flash presentations, we'll pause for a moment and take up some questions if they are available. And all the presenters, I request to be strict to the 4-minute time slot, whatever has been given to you. So I request Dr. Gabardhan Reddy from Jagi Vemana University to present the fast presentation. So we are going to take the control on your slides. So we'll be flashing the slides and control it.
Okay, thank you.
Yeah, please.
Thank you for your kind introduction about me. Before going to present our work, I would like to thank the organizers who are giving me opportunity to present our research work in our esteemed symposium. So next slide, please. So since the 10 years we are working on synthetic organic chemistry, particularly on the synthesis of metal N-nitrocyclic carbenes, which are used in various applications, CC and CN bond formations as well as CH relations. So there is our aim to publish various CC, CN bond and CR relation reactions. In order to that, we are prepared various N-heterocyclic carbenes, palladium as well as copper N-heterocyclic carbenes. So those applications, especially this Buchwald-Artig combination reactions and the Suzuki-Miura coupling reactions, which are we prepared our own catalyst and tested the catalytic activity and proven well in order to get the good reactivity. So in this motivation, we used to prepare some kind of copper N-heterocyclic carbenes from next slide, please. So the kind of benzene salts and the copper iodide. So we used to get we are expecting to get benzene copper N-heterocyclic carbene, but when you check the NMR and the crystal data, instead of copper N-heterocyclic carbenes, so we used to get such kind of C double bond O benzene 2 ones. So this is the motivation to prepare our benzimidazole 2 once. Actually, we thought to prepare copper N2-cyclic carbons. So, but initially, we are getting this kind of benzimidazole 2 once. So that's what then we moved to prepare various benzimidazole 2 once. So let us see the activity of various benzimidazole 2 ones, especially these are all very good biological receptors, some kind of anti-malarial, antibacterial, and also anti-HIV inhibitors. So when you check the literature reports previously reported by some other research group on this kind of benzimidazole 2 ones. So because of this reason, so we have to we switch over to prepare benzimidazole 2 ones instead of copper N2-cyclic carbons. We are going to continue to prepare those benzimidazole 2 ones. Next slide, please. So here some of the, so when you check the literature availability, where there is only our literature or any other. So there is not at all, there is no new compounds. But anyway, when we check the methods, what the methods are available so far for the preparation of benzimidazole 2 ones, when I when you see this Sun et al. Reported benzimidazole 2 ones from 1,2-phenylenediamine and pasogene. So here they used pasogene and also some other reagent. So then they prepared cyclized benzimidazole 2 ones. So here they used as cyclizing reagent as a pasogene. So some other group, this Ernst et al. Reported benzimidazole 2 ones by another method. So 1,2-dihaloxybenzene and ureas in presence of palladium catalyst. Here they are used some ligands, imidazole, and also some other Betty pass as a ligand. So here also there is another method to prepare benzimidazole 2 ones. Here they used pasogene. Here they used some kind of palladium catalyst as well as ligand, and also used some various conditions to prepare benzimidazole 2 ones. So other report also is available in unpublished in their in journals in 2013. They used copper iodide and tetrabutyl hydrogen peroxide as a oxidant. Here, oxidation reaction with some benzimidazole salts to benzimidazole 2 ones in presence of oxidant and metal precursor at a higher temperature. When you see the temperature at 120 degrees centigrade. So when you check all the reaction conditions, so of course we are also prepared some kind of benzimidazole ones. When you check the novelty of our reactions, since they are used, please next slide. Please next slide. So since there are all the reports, so they are used pasogene and some other ligands, some other additives, some other temperature. So this most of the above methods employed as a toxic. So when you talk about pasogene, it is a toxic metal. So when conduct some of the explosive materials and also used additives also used some tetrabutyl carboxylic acid as a additive, some of the reactions. So in our work, we used to, here also we used the oxidation reaction of benzimidazole salts in presence of our own palladium N2-cyclic carbene as a catalyst. Here, what is the difference between the previous reports and our reports? So since there are previous reports, they used some pasogene and some other additives and also used it to prepare. But here we didn't use any additive, and also we didn't use any kind of oxidant. Just only here we are using open air. So in the previous reactions, they are using tetrabutyl hydrogen peroxide, some other oxidants for the preparation of benzimidazole 2 ones. Just simply, we prepared benzimidazolium salts by orthophenyldiamine. Orthophenyldiamine, when react with aryl alkyl halides in presence of base, we are getting N-substituted amines. Subsequent reaction with the triethyl orthoformate, we are generating a series of benzimidazole salts. There, this benzimidazole salt can use it as a starting material. Then oxidation of these benzimidazole salts in presence of our lab palladium NHC. So we prepared a library of palladium N2-cyclic carbons in our lab for CC and CN bond formation. So then, based on our experience, so we started.
Dr. Reddy, Reddy, please conclude. You have crossed the time.
So when you optimize the conditions of this one, when you try with various catalysts and optimize the conditions, copper iodide and additives. So finally, when you see the various optimum conditions, palladium N-heterocyclic carbene and just open it, we got good results, and also just only 3 to 4 hours we got the product. And also this temperature is also enough, 80 degrees, and solvent is also, we need not use any DMSO or DMS, just only we use tetrahydrofuran as a solvent. So please next slide. Here we prepared various library of compounds by changing the NN substituents with benzyl to acetyl, acetyl. Here also we changed the substituent, instead of simple benzyl we are using substituted, and also electron withdrawing group like fluorine and other. And so this.
So Dr. Reddy, I request you to conclude.
Yeah.
It is good.
Yeah, this is it is concluded by NMR spectrum also, and also we concluded by single crystal data of those prepared compounds. So in summary, we prepared various benzimidazole 2-1s in presence of palladium N-heterocyclic carbene catalyst and also at higher temperature. So I thanks to IIT Tirupati for giving me chance to present our work in our esteemed symposium. Also thanks to Yogavan University Department of Chemistry to allow me present here. Also, finally, thanks to Council of Scientific and Industrial Research for financial support. Once again, I thanks to organizers for giving me this opportunity. If any queries, please chat me. I will happily answer. Thank you, Vanadal.
Yeah. Thank you, Dr. Reddy. Yeah.
Yeah,
So,
sorry to interrupt. I think we have to maintain time. So
Yes.
if there are too many slides, you go to the concluding one. And I request all the flash presenters, please, you should be able to adjust your time and explain. So I think please kindly follow this. Everyone, I request please follow that 4 minutes time. Okay, so you can always go to the final slide and explain. Yeah, all right, go ahead.
so next, may I request Mr. Sandeep Das from IISA Tirupati to present this your talk.
Am I audible, sir?
Yes.
Okay. So good morning, everyone. I am Sandeep Das from IISA Tirupati, and today's my talk is based on nitric oxide monooxygenation reaction of cobalt nitrosyl complex to cobalt nitritol base induced hydrogen gas evolution. Next, please. So, what is NOM? Basically, in biological system at higher pH, iron at first reacts with NO, leads to generation of iron NO plus complex, which further reacts with hydroxide, leads to generation of N-bound nitrous acid intermediate, which further releases NO2 and H plus to generate iron 2. That iron 2 further reacts with NO to generate iron 2 NO complex. Next. Next. So why NAR and what is important? Basically, in biological system, it is proved that NAR acts as a major source of nitrite. However, it is also reported that there might be some other source of nitrite, and NO may be converted to nitrite in biological system. In this regard, we choose to study nitric acid monooxygen reaction, and we took a well-characterized cobalt nitrosyl complex, which we prepared in our lab, and the nitrosyl is bent nitrosyl and further incorporated in the NOM reactivity. Next, please. So the reaction of the cobalt NO complex in a UV visible spectra, the characteristic band of 370 nanometer is disappear readily when the hydroxide added. Next, please. Analyzing the reaction mixture in IR spectra gives a peak at 1271, which shifted to 1245, which belongs to nitrite region. Next, please. Further mass analysis confirms that it is a cobalt nitrite complex, as it when we perform the reaction with the labeled NO, it shifts to 1 unit, which suggests that the cobalt nitrite complex is forming. So, what is the mechanism? Basically, from next, please. From literature survey, it is known that metal NO on reaction with hydroxide lead to generation of metal NO2 along with H2O. So from the previous literature and our assumption, we propose two types of reaction mechanism. That is, OH minus at first reacts with NO to generate N-bound nitric acid, which further undergoes homolytic or heterolytic cleavage. So in this regard, we should mention that homolytic cleavage required to one equivalent of base, and heterolytic cleavage required two equivalent of base. In this regard, we perform a titration where surprisingly you observe that the reaction is completing with one equivalent of base, which supports our assumption that the reaction is going on by homolytic cleavage, which further support by 2,4 DTBP test, which gives dimer as a single product, further confirmed by H2 gas evolution as to activate hydrogen recombined to generate H2. Further, our assumption confirmed by X-ray analysis, where we can see that one BF4 is present outside the coordinate pair to balance the charge, and it is a cobalt to NO2 complex. Next, please. If we summarize our work, then we can see that this is the first ever, this is an alternate pathway of conversion of cobalt NO to cobalt NO2. Apart from this, if you compare this study with the NIR study where same type of intermediate is involved, we can see that O-bound nitric acid can lead to generation of O-N bond homoerotic cleavage, whereas N-bound nitric acid can lead to generation homoerotic cleavage of OH bond. In NOM chemistry, basically H2 gas is forming, whereas in our chemistry, H2O2 is H2O it is basically forming. Apart from this, it opens up pathway of base catalyzed CH bond functionalization. And initially, it was believed that the only linear NO, that is NO plus, undergoes such type of electrophilic reaction. But this is the first ever report where we observed that the bent nitrosyl can also undergo such type of reaction. Apart from this, oxide gives same type of reactivity, and water is inert to such type of reaction. Next slide, please. So I would like to acknowledge my supervisor, Dr. Pankaj Kumar Kohli, and my lab members, and I would also like to acknowledge Ajay Tirupati and ACRB for the fellowship and instrumentation. And I would also like to thank IIT Tirupati for giving me a platform to present my work. Thank you for your kind attention.
Yeah, thank you, Mr. Sandeep, to make it within time. So next, may I request Mr. Selva Kumar from IIT Madras. Mr. Selva Kumar from IIT Madras.
Yes, sir. Can you hearing my voice?
Yes, yes.
Yeah.
Please go on.
Yes, sir. Good morning to all. First of all, I thank to IIT Tirupati giving the opportunity. Myself, Selva Kumar Armagam, under guidance with the Karthik Chandra Medical Department of Chemistry, IIT Madras. I would like to give my presentation here, recent publication manuscript. My title, Redox Active Exonuclear Mixed Valency Dicationic Cerium 3 plus Cerium 4 plus Coordination Cluster. Please go next slide. Yeah. We are reporting here first time mixed valency cerium 3 plus cerium 4 plus reaction and mechanism studied by HRMS and as well as reaction and mechanism of alcohol oxidation. Successfully, we isolated intermediate cerium 4 dicationic PCS. This is the third mixed valency cluster and rarely documented so far. So we started synthesis of the metal cluster. The cerium nitride hexahydrate with skip-base ligand under basic and aerobic condition. We have synthesized 3 mixed valency metal cluster and 2 different counter ions depend upon solvent medium, methanol and acetonitrile. Next slide, sir. We look at see here left side cerium 4 plus and final product cerium 6 dicationic mixed valency dicationic species. You look at the left side, each cerium atom bridged to Me4 oxygen atom and cerium 3 plus oxidation state confirmed by XPS method. That right side, the final product mixed valency cerium 4 plus we confirmed by single crystal XRD, as well as is below C the counter ion in the cerium same cerium counter ion. Next slide. So we characterized by that metal cluster. So first we theoretically determined by oxidation state PVS calculation and experimentally photoelectron spectroscopy. We see here absorption friend and 3 absorption friend, cerium 3d5/2 and 880 electron volt and 900 electron volt. It is corresponding to +3 oxidation state, and 914 electron volt is corresponding to +4 oxidation state. So we confirmed that our metal cluster have both oxidation state. The metal cluster redox property confirmed by cyclic voltammetry. 1 electron QC reversible oxidation process was observed. And one more thing, we want to know this metal cluster stable or not. So we studied in the HRMS solution state. In the molecular species isotopic pattern and simulated pattern, both are exactly matching. So we confirmed this metal cluster in the stable in the solution state. Then we studied the magnetic susceptibility in the DC field 0.5 Tesla, 300 Kelvin to 500 Kelvin. we see, look at it here, chi t versus t plat, 300 Kelvin to and 5 Kelvin. It's slowly decreasing the chi t value because depopulation of the star effect in the cerium and cerium ions. So we weakly antiferromagnetic coupled to this mixed valence cerium cluster. So next slide, sir. And then we studied the reaction and the mechanism by using the HRMS. I already mentioned previously. So 0 to 72 hours is the time dependent HRMS we measured. So we found in the intermediate cerium 4 fast in the HRMS. Then we crystallized and successfully crystallized and measured the single crystal XRD structure. Then the final product cerium 6 also we found, and so each simulated pattern and experimental pattern both matching, so we confirmed cerium 4 and cerium 6 cluster. Next slide, sir. Then we found we have found application of this metal crystal alcohol oxidation in the benzyl alcohol. So reaction mechanism is studied by HRMS as well as UV. So we optimize this benzyl alcohol in different conditions, and one of the metal cluster and reaction with benzyl alcohol, we took reaction solution in the HRMS. So the one of the aldehyde alcohol is binding to the cerium cluster. So we confirmed the HRMS. So we proposed the reaction and the mechanism. So the phenyl radical intermediate forming. So my conclusions: we have first reported mixed valency cerium cluster studied by HRMS and successfully
Selva
intermediate.
Kumar, may I request you to conclude?
Yes, sir. My final conclusion: we have first reported mixed valency cerium cluster HRMS studies and successfully crystallized the intermediate species and tempo-free alcohol oxidation. Acknowledgement: I would like to express special thank to my guide Karthik Chandra Mandel and my lab mate CSR Fellowship and IIT Madras, ISER Tirupati for instrumental facility. Thank you for your kind attention.
Thank you, Mr. Selva Kumar. Yeah. So next, may I request Dr. Rahul Nag to come and present your talk.
Am I audible?
Yes, yes. Please go on.
Yeah. Good afternoon, everyone. I'm Rahul. I would like to present one of my PhD work titled Spiroindolene Functionalized Calyx Fluorine, Copper Binding and Cancer Cell Diagnostic, which I did working under Professor C. P. Rao. Next slide, please. Yeah, starting with the relevance of the spiroindolene based system, due to its ring opening nature and stimuli responsiveness, it has been used in metal ion sensing, drug delivery vehicle, as well as targeting cell organelles. Please go to the next slide. Yeah. Working with the calixarine based systems, we have functionalized the lower rim of calixarine with the spiroindolene moiety to give a multi-functional nature. And we have characterized the molecule using spectroscopy, NMR, and NIMS. Please go to the next slide. Yeah. So what we have done, we have studied the metal binding of our ligand, and we have used copper. Why have we have used copper? For its biological applicability of different of its complexes in the including anti-cancer activity. So first we have studied the spectroscopy using absorption and fluorescence study. So what we have found, we found ratiometric binding along with fluorescence quenching in presence of the copper ion. And when we did ITC, we have found one-side binding or 1:1 binding, which we have also found from DFT study, where after ring opening, copper ion binds in the lower rim of calixarine using distorted tetrahedral geometry, using spiroindolene and triazole moiety. We have also visualized our molecule and the complexes using microscopy. We have found from SEM that the ligand alone does not have any structure, while the copper complex in situ or the isolated both have rod-like or crystalline shape. Please go to the next slide. So, as I already said, due to the biological applicability of the copper complex of different organic systems, we have done anti-cancer activity of our ligand as well as the complex using MD MB231 cancer cell line, and found out that though the ligand does not have any cancer cell killing potency. The copper complex have very high potency and it has a IC50 value of 165 nanomolar. So when we wanted to study the cancer cell killing mechanism, we found out that sub-G1 population increasing with the increasing concentration of our sample along with the decrease of G0, G1 and G2N population. And we have found the cell internalization using ICP where we have shown increasing copper ion concentration in the cell as we increase the concentration of the incubated sample. Please go to the next slide. So as we already discussed the fluorescence studies and the quenching in presence of copper, we did confocal microscopy in the cancer cell. We found the ligand showed red fluorescence and which is quenched in presence of the copper complex. We did colocalization with microdata green where we found orangish yellow color and overlap integral of 0.9. Though the copper complex did show quenching of the fluorescence, we could see rounded cell morphology, which is an indication of dead cell, which we have already shown earlier through MTT assay. So we have generated an cancer cell theranostic system where ligand is showing good cell imaging and the complex is showing cancer cell. Please go to the next slide. Finally, I would like to acknowledge by first acknowledging my Professor C.P. Rao as well as IIT Bombay for all the facility and IIT Tirupati and the conference organizing committee for giving me this opportunity. Thank you.
Yeah, thank you, Dr. Rahul, to make it very well within the time. Thank you so much. So next, may I request Mr. Mahesh Enuganti from ISA Tirupati.
Sir, am I audible, sir?
Yes, yes. Please go on.
Yes. Good morning, all. Today I'm going to talk about NOD reactions. That is a nitric oxide dioxygenation chemistry of bio-inspired metal oxygen adducts versus metal nitrosyl complex. Next slide, please. Yeah. Before 1990, nitric oxide is a toxic molecule, but after 1990, a Nobel Prize were awarded by J. Egnaro et al. In physiology. So after that, nitric oxide is very good essential molecule for signaling as well as neurotransmission, and it acts as a vasodilator and etc. After that, sorry, hello, yeah, can you back, please? Yes. If you have excess nitric oxide in your biological system, then it is a problematic. So for that, who will put the nitric oxide levels as a biotolerance level, that is 25 to 35 ppb level? There is an enzyme is there, that is nitric oxide dioxygenase enzyme. What it does, it converts the toxic nitric oxide gas molecule into a biological benign nitrate ions. So the mechanism is initially the metal complexes reacts with oxygen molecules and it forms a metal dioxygen species, or otherwise metal complexes reacts with nitric oxide and it forms a metal nitrosyl species. So after that, these metal nitrosyl and dioxygen species in the presence of NOD enzyme, it converts into a nitrate ion. Next slide, please. So in this way, we are also contributed small NOD chemistry by using a cobalt and nickel peroxo metal complexes having a same ligand structure, that is a tetramethylated cyclone ligands. Next slide, please. So in coming to the first NOD chemistry, that is cobalt peroxo complexes reacts with nitric oxide gas at minus 40 degrees centigrade under argon atmosphere. It forms a cobalt nitrite species. So in UV spectra, we found a new peak at 535 nanometers. With time, it decreases, and in IR spectra, we observed a new peak at 1271 centimeter inverse. It is already literature reports, it is showing this is a nitrite stretching frequency. So after that, we examine ESIMS analysis, and there also we got 331.1 MIZ value. It corresponds to a cobalt nitrite species. So after that, we did labeling experiment. In labeling experiment, we got 333.1 MIZ value. It corresponds to a metal nitrate with labeled oxygen. Next slide, please. So far, the NOD chemistry is reported only from metal superoxo or peroxo to NOD products, that is metal nitrate or nitrates. But after that, the completion of NOD cycle didn't reported still yet. So in that way, we did some NOD enzymatic cycle reactions here. So for that we did the from metal nitride to metal peroxo complexes, and also from metal peroxo complex to metal nitrosyl complexes. So we successfully did these reactions. So we observed the metal peroxo complexes from NOD products, and from metal peroxo NOD products as well as from the initial metal peroxo complexes we observed the metal nitrosyl complexes formation also. Here in UV visible spectra, 350 nanometers peak is corresponds to a metal peroxo complexes, and this 370 nanometer peak is corresponds to a metal nitrosyl complexes. In proton NMR spectra, we confirmed this is a these metal nitrates are D7 system having spin at 3/2, and this initial after reaction with nitric oxide, it turns to its oxidation state changes from +2 to +3, and it is a D6 system and diamagnetic species. That's why it is showing the NMR active. Whereas in the first case, it is a paramagnetic species. That's why it doesn't show any NMR peaks. So next slide, please. So after coming to the nickel nickel NOD chemistry, we prepared nickel peroxo complexes, and after preparing these nickel peroxo complexes, we purged the nitric oxide gas under at -40 degrees centigrade under argon atmosphere. So in these reactions, we observed a new peak formation at 350 nanometers. With time, it decreases and it gives a new peak at 585 nanometers peak. So these 350 peak formation were observed in only at -40 degrees centigrade. This is the formations peak, and if you did the same reaction in room temperature condition, we did not observe any new peak at 350 nanometers. So this reaction is via progressed intermediate, that is peroxynitrite. So this peroxynitrite is very much unstable at go back to next slide, please. Previous slide, please. Yes. So at room temperature condition, we did not observe any intermediate.
You need to conclude.
Yes, sir. One second. Next slide, please. So for completing the NOD's enzymatic cycle, interestingly, it shows the metal peroxo species formation, but it did not shows any metal nitrosyl formation at low temperature as well as in room temperature conditions. Next slide, please. So while summary, here I am showing the metal peroxo complexes having the same oxidation state with same ligand framework, shows the nitric oxide deoxygenation chemistry, but whatever the NOD products were formed, those were showing the different activity with nitric oxide, but those were shows the same activity with dioxygen activation. So here I acknowledged Department of Chemistry and Dr. Pankaj Kumar Kohli, my supervisor, and my lab members, and Department of Chemistry for giving this IIT Tirupati for giving this great opportunity to present my work in this NSRTCS. Thank you.
Thank you, Mr. Mahesh. Thank you so much.
Thank you, sir.
May I request presenters, if possible, you can turn on your video because it is getting recorded. So that will be really nice if you turn on your video when you are presenting. So next, may I request Ms. Vasavi Dutt from SRM University, Andhra Pradesh.
Sir, am I audible, sir?
Yes, yes.
Good noon, everyone. I am Vijay Vasavi from Department of Chemistry, SRM University, Andhra Pradesh. Firstly, I want to thank NSRTCS 2020 team for giving me this opportunity. Here I'm going to talk about one of my recent works, tunable emission in perovskite nanocrystals with simple hydrohalic acids. Next slide, sir. Yes. So perovskite are the materials which are having the crystal structure ABX3, and metal halide perovskites are one of the category. And we all are familiar with a thin film based metal halide perovskite, which is widely used in solar cell application. Their colloidal counterparts is also very promising because of their intriguing optical and charge transport properties. They were first synthesized in 2015 by Professor Kovalenko using hot injection synthesis approach. This approach typically involves some of the degassing and nitrogen atmosphere. So from the HRTEM image, we can say that this material is highly crystalline. And if we look into the optical properties, we could achieve the spectral tunability from blue to all the way red by varying the halide composition. Many researchers across the globe try to achieve this using various halide sources like Grignard reagents, organometallic halides, which are pyrophoric and which also involve some complicated processing steps. My motivation is to get the spectral tunability by anion exchange using metal halide salts via simple post-synthetic modification where the control conditions are not required. Next slide, sir. So in my lab, I have synthesized the cesium-lead bromide perovskite nanocrystals in open atmospheric conditions. Still, I could achieve a good photoluminescent material, which is exhibiting the photoluminescence quantum yield of over 60%. We can see in the photographs which are taken in normal light conditions and with the irradiation of UV light. So here shows the TEM image of the cesium-lead bromide perovskite nanocrystals. From this, we can say that they're highly monodispersed. And from the X-ray diffraction pattern, we observe that this material is crystallizing in cubic phase and followed by the optical properties as shown here with a large absorption cross sections and narrow emissions. Next slide, sir. So as I already discussed earlier, my main motivation is to achieve the anion exchange using simple processes. Initially, I have synthesized the cesium-lead bromide perovskite nanocrystals, and I did a simple post-synthesis treatment using hydrohalic acids. With addition of hydrohalic acid, I could achieve cesium-lead chloride nanocrystal, which is giving blue emission as shown in the photograph. And further, with the addition of hydrophobic acid, I could achieve the back exchange. I mean, the cesium-lead bromide. I could retain the PL properties to a maximum extent without compromising the degradation of PL properties. So here shows the time relied blue shift from 510 nanometer to 430 nanometer with chloride exchange, and a red shift from 430 nanometer to 510 nanometer with back exchange. Next slide, sir. Coming to the structural properties, here shows the transmission electron microscope images of the parental cesium-lead bromide perovskite nanocrystals, followed by the cesium-lead chloride nanocrystals obtained by chloride exchange, and the cesium-lead bromide obtained with back exchange. Through the size histograms which are represented here, we can say that this material is highly monodispersed and the particle size is largely maintained. So there is a small decrease in the particle size of cesium-lead chloride perovskite nanocrystals, which is attributed to the fact that of the ionic radii difference of bromide ion and chloride ion, which changes the interplanar spacing in the lattice and gives the overall shrinkage in the crystal. So from the X-ray diffraction patterns, we can say that these are highly crystalline, and also they are in good agreement with the standard database JCPDS, and we observe that they are crystallized in orthorhombic and cubic phases as well. So next slide, sir. So not only the hydrohalic acids, I have used various other metal chloride salts like sodium chloride, potassium chloride, magnesium chloride, ammonium chloride to get the spectral tunability from green region to blue region. So using a simple post-synthetic modification, which is everything done in open atmospheric conditions, and I could achieve a good quality cesium-lead halide perovskite nanocrystals, which can be further employed for light-emitting applications. This work has been recently published in Pristine.com. And finally, I want to acknowledge SRM University, Andhra Pradesh for all the support, my supervisor, Dr. Nimai Mishra, and my co-workers for their guidance and motivation. Thank you for your time.
Thank you, Ms. Vasavi, for making it within time. Thank you so much. So next, may I invite M. Nikita from Sri Padmavati Mahila Vishwavidyalaya.
Am I audible, sir?
Yes, yes, yes. Please go on.
Yeah. Yes, thank you. A very good afternoon to one and all present in the symposium today. I'm Nikita and I will be presenting on the fissile co-precipitation synthesis and characterization of the Fe3O4 nanoparticles. Next slide, sir. Magnetic nanomaterials have opened a new avenue in the field of medicine today because they can be easily manipulated by external magnetic field, which can be used in drug targeting. Hysteresis and other losses which occur in alternating magnetic fields can be applied in hypothermia applications. Their ability to extract high energy, their intrusion deeply into tumor tissues, and their effectively way of crossing the blood-brain barrier can be have been found very useful in the field of medicine today. Next slide, sir. Falling under this category, Fe3O4 nanoparticles being stable, biocompatible, minimally toxic with large saturation magnetization value of around 90 EMU per gram are being used in magnetic hyperthermia based cancer therapy. So it is basically able to heat tumor cells above 43 degrees Celsius without damaging normal tissues because we know that cancer cells show more weakness to high temperatures compared to the ordinary cells. So basically, the tumors are fed with magnetic nanomaterials, and externally an alternating magnetic field is applied. And with this interaction, the nanoparticles are able to dissipate power and heat is generated. This is represented in the form of hysteresis loop, which can be seen below. So the power dissipation is directly proportional to the area under the loop. So if we need to get more power, we need to increase the area under this curve. Next slide, sir. So we have used the co-precipitation method to synthesize the PEG coated Fe3O4 nanoparticles. Basically use PEG for the better dispersibility and to improve the biological penetration. I won't be talking about much about the synthesis because it is well known. We have used this basically because it is easy, economical, and very good homogeneity is obtained in this method. Next slide, sir. The synthesized Fe3O4 nanoparticles were given for XRD analysis, and as seen, we have obtained single phase cubic structure nanoparticles, and the crystallized size was found to be about 8.2 nanometers. And the EDS spectra and the semi-images have shown the presence of both the elements oxygen and iron in their respective percentages, which were matching with the theoretical values. The crystallized size is yet to be confirmed with the TEM images, and we need to give the sample also for the VSM analysis, that is a vibrating sample magnetometer from where we actually get the hysteresis curve. Our research, we have just started this work, so it is still in the infant stage, and a lot of characterizations are yet to be done. Next slide, sir. I would like to present the future coursework or the plan of actions that we are going to take upon this. The magnetic hyperthermia has been proposed in the year around 1957, but till now it is still under preclinical stage only. So the main challenge is to attain high specific power loss, that is to increase the area under the hysteresis loop. Currently reported value is only around 65 EMU per G, but the value we can take is up to 100 to 200, and also we need to increase the specific absorption rate. All these things have to be done keeping in mind the field and frequency which are safe for human body, and we need to make sure that the particles that we synthesize are biocompatible with low toxicity. And one of the major challenges is to maintain stable temperature at the target sites. So we are looking at optimizing parameters like size, shape, and surfactants, and also use various experimental techniques to see how they affect their size. For example, now we have used the co-precipitation method. We are also looking at hydrothermal method, how it is going to affect the size. And much work has not been done into tuning the magnetic anisotropy. This can be done by alloying various magnetically soft materials with magnetically hard materials. To put it in easy words, I could say that now we have only Fe3O4, so it is about doping it with cobalt or nickel or manganese, any such few things. So talking about the recent trends, I cannot but talk about the artificial intelligence. Few papers have also told that, they have been training the models with the available data so that parameters can be predicted and they can be used. So I can say that we have just started out, and this is a very interdisciplinary project. We need to look at biological aspects and also clinical aspects of this. And I would tell that magnetic hyperthermia will definitely make its way prime time into curing the cancer more effectively and more economically if these things are improved. I would like to thank my supervisor, Dr. P. Subhashini, and the DST Curie lab at our university for providing amenities, and the organizing committee of the symposium for giving me this opportunity. Thank you so much.
Yeah, thank you, Nikita. Thank you so much. Yeah. So next, may I request Gunashekar from VIT, Vellore Institute of Technology, to present your work.
Yes, sir. May I audible, sir?
Yeah, you are audible. Please go on.
Hello?
Yes,
okay,
you
sir.
are
Very
audible.
good afternoon to everyone who have presented. Yes, sir. Yes, sir. Very good afternoon to everyone. And thanks for the team NSRTC who are giving me this opportunity to present my this work. Coming to my work, synthesis of aromatic hyperplanchal polyester and the fabrication of piezoelectric sensor based on electrospun meet PVDF and its blends with aromatic HTP. Coming to PVDF, we all know that PVDF has a high crystal phase, and piezoelectric sensors are the main application for us. Coming to this synthesis, we use diphenolic acid and pentoalkyl as a core and monomer molecule in the presence of para-toluene sulfuric acid as a catalyst. Under nitrogen atmosphere, we get this idealized hyperbranch polymer structure. After this hyperbranch polymer synthesis, we do our interest is to do electrospinning and to generate nanofibers. In this electrospinning technique is very best technique to get nanofibers, in which we can enormously increase the beta phase of a PVDF when we add this hyperbranched polymer compound. Now coming to electrospinning, we can see a flowchart here regarding that electrospinning, what parameters we have maintained. Initially, we mixed PVDF with aromatic PVDF, aromatic HPP in the presence of 6 ml DMF and 4 ml acetone solution. Later, we get the clear solution after stirring for 24 hours. Later, that clear solution is used for electrospinning sample. After getting that electrospinning sample, we will do some characterizations like same time, FTIR, XRD, etc. to confirm the beta crystallinity increase in the PVDF HPP content. Later, we have done the nanoweb fabrication of this PVDF HPP sensors. We can see these images regarding that fabrication. This work is done in Korea, Kyunghee University lab. And coming to results and discussion, if you see that figure 2, we can see the first figure, which is regarding the, hello?
Yes, go ahead.
So we can see that PVDF, which is giving 0.95 peak-to-peak voltage value under the presence of 1 Hz frequency and 1 kg of load. Later, if we add PVDF along with HPP in the 90-10 ratio, we can get a VPP value which is enormously increasing when compared to neat PVDF. Here we can say that by adding this HPP, we can increase the beta crescent phase, and we can also increase that electric character of that sensor. Later, our interest, we move on to changing varying frequency as well as varying load. Now we can see these 3 images in figure 3, which is varying frequency 0.1 Hz, 0.5 Hz and 1 Hz. Here what we observe that when we increase the frequency, the VPP value decreases. This is based on the relaxation time. Then the frequency is very less, relaxation time is very more. Here we get the signal VPP value more. If we increase the frequency, the VPP value will decreases gradually. Next slide. Here, next in figure 5, we increase the load by maintaining the 1 Hz as a constant frequency. Here we can observe a trend that while increasing the load from 1 kgf, 2 kgf and 3 kgf, while increasing the load, the VPP value is gradually increasing. This is concluded in the below figures. We can see the quantitative values of the frequency as well as load here. While frequency is increasing, we can see the VPP value is gradually decreasing. Here, if we come to this load value, while load value is increasing, it is gradually increasing. Next slide. Now coming to conclusion, we have electrospinning of neat PVDF along with its blends of aromatic HPP, which is synthesized in our lab, was carried out and measured for piezoelectric behavior under 1 kg of load and frequency. Later, PVDF HPP sample exhibited the highest output signal compared to neat PVDF. Later, we done the work along with increasing load as well as increasing frequency. Here, in case of increasing load, we can see the decreasing frequency. We can see the 1 kg to 3 kg of VPP value is increasing. Later, frequency is decreasing from 1 to 0.1 Hz. VPP value is increasing. This piezoelectric data obtained from this work can serve as a evidence that PVDF HPP even 9010 fiber can be effectively used as an active material in the piezoelectric sensors and energy harvesting applications. So see these are 3 images which is regarding the electrospinning and piezoelectric measurements, simple circuit diagram and analyzing data. This is mainly used for the health monitoring applications. Coming to acknowledgement, my guide wish to thank CSIR Government of India for supporting our work and also VIT for providing VIT seed grant for carrying out this work. Thank you.
Thank you, Mr. Gunashekar. Thank you so much.
Thank you.
So we'll pause here for a moment and let's see if our audience has questions, comment, or suggestions. Okay. So the stage is open for any questions, comments, and suggestions. So all the participants, if you have any questions or suggestions, so please feel free to bring it to our.
Yeah, Arun, keep it only for 5 minutes total.
Okay. Yeah. Is there any questions?
Yeah, I just can you hear me?
Yes, Professor Rangarao. Yeah.
Yeah, I just the previous speaker, that iron particles.
Yes, sir. Yes, sir. Good
Which
afternoon, sir.
institute are you coming from?
Sir, from Sri Padmavati Mahila Vishwavidyalaya, sir, Tirupati, sir.
Oh, very good. Okay, very good, very good. That iron particles, once you prepared, you have this XRD taken there, right?
Yes, sir. Yes, sir.
XRD is very broad.
Okay, sir.
Do you how did you use that to estimate the size of the particles?
Uh, Sir, broad in the sense, I mean, they are just, I mean, to notice the angle rightly, sir. That is why, I mean, we have got the angle correctly at the where we have to get the peaks. And the crystallite size was calculated using the Debye formula, sir. We have taken the angle at where the peak was obtained, and the angle was substituted along with the parameters like the wavelength used in the XRD and the full width half maximum value that was obtained from the graph itself. And using these values, we have calculated the crystal size, sir.
Oh, very good. So you have taken only one peak into consideration, not multiple peaks. The intense peak, I think you have taken into consideration. Is that right?
Yes, sir. That is the main character. At 311, we have got that.
Okay. Good. Very good. Very good. Thank you.
Thank you. Thank you, sir.
Is there
Thank you, sir.
Is there any other questions? So if you have any other questions for other participants, please come forward.
I have one question for
Yes,
Kumar
yes.
Armugam, IIT Madras.
Okay, please.
Sir, could you
Yes, sir.
please how much difference in oxidation reduction potential of ceria when it is in parental form and then in the metal cluster form? Is there a big difference or?
Electron potential minus 0.11 volts. Sir. So as previously reported, cerium 4 and cerium 3 plus one oxidation reduction potential. Yes, sir.
Okay.
So is there any other comment or suggestion?
I have one query for this person Mahesh from Iser Tirupati, I think, who presented an NO.
Yes, sir.
Yeah. So in your case, you have that peroxo and then treated with NO, and then you're getting NO2, right? So
in one case, NO2, sir, cobalt peroxide with nitric oxide, we are getting cobalt nitrite, NO2. But in nickel case, we are getting NO3.
NO3. Okay. So in NO2 case, which is the oxygen that is going out?
Sir, actually, we trapped that oxygen atom also by using triphenyl phosphine, and we observed a peak in GCMS formation of triphenyl phosphine oxide.
Okay, the O which is going, is it from NO or is it from peroxo?
So no, from peroxo, sir. Actually, we labeled also that experiment. With labeled also, we confirm that oxygen is coming from peroxo.
Good.
Yes,
okay,
sir.
Thank you.
We have time to take one more questions or suggestions or comments if you have.
Okay, I ask one question.
Okay, the comment I would like to make.
Yes.
Is
Professor.
that the flash presentations yesterday and today are all very good.
Great.
We could have probably there's not much time, I guess, but we could have given 1 or 2 minutes more. It also gives them an opportunity to present the work in a short form quickly. I think that's a good exercise that is designed in this symposium. I have to congratulate all the flash presenters, young students, and then from different institutes.
Thank you, Professor.
Exactly. Professor Rangarao, I just want to say one thing. Yes, we could have given 1 or 2 more minutes, then we have to cut number of people. So what we thought was we can encourage more number of youngsters so that they can come up in future.
That is true.
That was the idea.
Actually,
that was the idea.
Actually, I would have asked my students to go through this exercise. I didn't know that.
Sure. Okay.
I thought only you are giving us to present our work, but I could have asked my students. It's a test for them to present in few minutes what they have done. That's a good thing.
Sure. In the next symposium, they'll be there. Okay.
Thank you, Professor Rangarao, for the feedbacks. And in coming days, we'll be having many more symposium and conferences. We'll take all these into account. Thank you so much. So it's time to move on. So may I request Mr. Sai Manoj from IIT Madras to present your work?
Yes, sir. Am I audible, sir?
Yeah, you are audible.
Yeah, thank you for NCRS TCS for giving me this opportunity. I'm Sai Manoj, working with Dr. Karthik Chandra Mandal, IIT Madras. I would like to speak about my recent paper on stabilization of linear C3 moiety by two donor ligands, a theoretical study. So using triphenyl phosphine and heterocyclic carbene and cyclic alkyl carbene. The main, next slide please. Next slide please.
It's there already.
Yes, please.
Next to this?
Yeah, no, back. Yeah, this slide. Yes. So here the main aim of the study to understand the bonding between the central C3 unit and the ligands. So the theoretical optimization at the BP86 level shows that the C3 stabilized by triphenyl phosphine unexpectedly has a synclinical gauche conformation compared to the trans conformation as originally suggested, whereas N-heterocyclic carbene has a transband geometry and the cyclic alkyl carbene has favors a linear geometry. So here, and the main the choice of ligands is based on the pi accepting capacity, and here we can see that the CAC stabilized cyclic alkyl carbene stabilized C3 has more dissociation energy and is thermodynamically more stable compared to the N-heterocyclic carbene or cyclic triphenyl phosphine stabilized C3. The NBO analysis shows that the PC bond here, the PC bond is it's a single bond with a more polarized towards the C3, more polarized towards the C3 unit, and whereas in case of N-heterocyclic carbene on cyclic alkyl carbene stabilized C3, the bond is more polarized towards the little bit slightly polarized towards the ligand, and it suggests a double bond character. Next slide, please. The true nature of the bond can be understood by using energy decomposition analysis, so which gives the intrinsic interaction energy between the two interacting fragments, which is mainly composed of 4 different fragments. So one is the electrostatic contribution, which takes into account the electrostatic interaction energy, and the delta E Pauli, which takes into account the Pauli repulsion between the interacting fragments, and delta E orbital, which takes into account the orbital interactions, and delta E dispersion, which takes into account the dispersion energy. So for the bonding study, I took two different units like for the study. So the two ligands as one fragment and the central C3 as another fragment. So to study the bonding on both sides of the C3 unit. So I've considered 4 different bonding possibilities to come to a conclusion that was the best bonding description. The first bonding possibility being the two interacting fragments in their singlet state, neutral singlet state, giving rise to dative bonding. And the second possibility is the two interacting fragments is a neutral state and electronic quintet state participating in electron sharing bonding. And the third possibility is the two interacting fragments as a singly charged species like L2 plus and C3 minus in electronic doublet state participating in the dative and electron sharing bonding. And the fourth possibility being the two fragments in their electronic a triplet state with a doubly charged species like L2 plus and C3 minus participate in the triplet state, participating in both electron and dative electron sharing and dative bonding possibilities. Next slide, please. So of these 4 bonding possibilities, the bonding possibility with the lowest delta U orbital is considered as the best bonding possibility, and here it comes to the conclusion that the bonding possibility with in the case of triphenyl phosphine and NHC, the dative and electron sharing possibility with interacting fragments in their singly charged species in the doublet state is the most feasible type of bonding, whereas in cyclic alkyl carbene, the best description of bonding comes from the interacting fragments in their electronic quintet state, giving rise to electron sharing bonds. So coming to the pairwise interactions of the best bonding interaction, best bonding possibility, here there are 5 pairwise contributions for PPH3 or stabilized C3 or NEC stabilized C3. So where the first contribution or the strong contribution to the delta orbital energy comes from the sigma donation from ligand to the C3, whereas the second contribution is coming from the sigma electron sharing between the ligands and C3, and the third, fourth, and fifth contributions are from the pi back donations from C3 to ligand. Here is a small, this is a pi back donation. And whereas in the case of cyclic alkyl carbene stabilized C3, there are 4 pairwise interactions. The first 2 are coming from the sigma contributions, electron sharing sigma contributions between the soma of the fragments, and the next 2 are coming from the pi contributions. Next slide, please. So Next slide, please. So here are the deformation density plots of these corresponding to the pairwise interaction. So here the red to blue is the charge transfer and charge migration, and here are the interacting fragments. So here we can see that the contribution from the contributions, the sigma contributions are always higher than the pi contributions, be it electron sharing or dative back donation. So sigma contributions are higher. Next slide, please. So, in conclusion, we can say that from these from the energy decomposition analysis and the natural bond orbital analysis, that the the higher pi accepting capacity of the CAC ligand favors the electron sharing bonds between the ligands and the central C3 unit, they're best described as a cubulin type of structure. Whereas in case of PPH3 or triphenylphosphine or any stabilized C3, it is a mixture of electron sharing and dating bonds between the ligands and the central C3 unit. So I thank Dr. Karthik Chandra Mandal, my supervisor, for giving me this challenging problem. And I thank Professor Gernot Franking and Dr. Sudeepan and for constant support and guidance. And I thank IIT Madras and I thank CSR for fellowship. Thank you very much.
Yeah, thank you, Mr. Sai Manoj, to make it within time. So thank you so much. So next, may I invite Mr. Devendra Reddy, so from IIT Tirupati. So Devendra, please come. Yeah, Devendra, please come.
Am I audible?
Yes.
Okay, sir. Today I am discussing theoretical spectroscopy of isotopic dilute water and hydrophobicity. Next slide, please. In this work, our aim is to study water structural changes near to the hydrophobic surface by using theoretical spectroscopic modeling and hydrogen bonding analysis. For this, we have used only methane water system methods. We have used classical molecular dynamics for water model SPCE, methane, OPL, SAA. For quantum calculation, we have used a hybrid functional B3LYB basis set. This one clusters selected from molecular dynamic trajectory. A water molecule selected from what we keep water molecule as center and a selected cluster, and that water molecule we have stretched that with we have stretched selected water molecule and performed DFT calculation. We got this potential energy surface diagram. After that, we solved Schrodinger equation and we calculated transition frequencies and dipole moment and salvation energy. Next slide, please. This is the spectroscopic maps and relations of these. We got these relations, binomial relations and fitting parameters. These black color in water structure, and remaining all color dots are methane water system. Next slide, please, sir. Each and every step of DFT calculation is difficult. That's why we have generated classical versus quantum salvation energy map. We got this relation from this classical salvation energy. We got this quantum salvation energy. After that, transient frequency and transient dipole moment. From this, we got linear AR spectra. From linear response function, real Fourier transform of linear response function. From this, we got the spectra. Here, spectra of bulk water and methane water. Methane water, small blue shift in presence of methane, and also more structural ordering compared to bulk water. Next, we have analyzed more here by using histogram method. Here in figure A, we compared bulk water and salvation cell-wise water and water facing methane. In figure B, compared with bulk water and second cell, third cell, almost similar to bulk water. In figure C, bulk water compared with first cell water. First cell water towards redshift compared to bulk water. Next, right side figure A, we have compared with water facing towards methane and high temperature water. Here, high temperature water facing methane behave like high temperature water. In figure C, we compared with first cell water with low temperature water. Here, first cell water molecules behave like low temperature water. Salvation cells are based on this RDF. Salvation cells are selected based on this RDF. Next slide, please. Here, further analysis, we have done hydrogen bond analysis. These are average hydrogen bonding in different solvation cells and bulk water. Here in this distribution, first solvation cell, broader distribution. That means some of the water molecules got more hydrogen bonding. Some of water molecules are less hydrogen bonding. Conclusion. Some of the water molecules was stretching frequency towards redshift in first solvation cell, and those are facing towards methane was stretching frequency towards blue shift. Water molecules facing towards methane behave like high temperature water. Some of the first cell water molecules behave like low temperature water. Reference for this work and acknowledgement. Thank to my advisor Rajiv Biswas sir and IIT Tirupati, and thank you to all.
Yeah, thank you, dear Devendra. Thank you so much to make it within time. So next, may I invite Mr. Kulbir from ISA Tirupati to present your talk. Mr. Kulbir.
Is he there or?
So, Mr. Kulbir.
He was here. He is here.
Please unmute your mic.
He is on unmute mode only, but we cannot hear. Okay.
Please
so
talk.
Mr. Kulbir, you please leave the meeting and again try to join and adjust your audio device. We will move for the next speaker.
So we'll take you at the end. Okay. We'll bring you at the end. So next, may I invite Mr. Albin Joy from IIT Tirupati.
Hello, good afternoon, everyone. Today, first of all, I thank the organizers of this symposium for giving me a chance to present my work. Today, I would like to present my studies on structural details of azurin to further understand its interaction with the tumor suppressor protein P53. So, in this work, we are studying the structural details of azurin, and in part, we will study its interaction with tumor suppressor protein P53. So here the structure of azurin is shown here. It has 8 beta strands fashioned in a beta barrel structure, and a copper ion is coordinated as type 1 copper center. The hydrophobic core of the 8 beta sheet is shown here. So first we employed replica exchange molecular dynamic simulation to understand the melting process of the melting temperature of the protein. So from the RMST and FASA calculations of the different replicas, we can see that there is a transition at around 400 Kelvin. So it is the azurin is a very stable metalloprotein. Moving to next slide. So further we analyze the denaturation of holoazurin and apoazurin. Holoazurin is the form of azurin containing the copper, and apoazurin doesn't contain the copper ion. So from the studies of RMSD and sample calculations, we can see that by 20 nanoseconds, holoazurin is unfolding, while apoazurin unfolds only above 30 nanoseconds. So apoazurin is found to be more stable than holoform. So one probable reason for this is the changes in the microscopic arrangement of atoms of the beta sheet residues in the hydrophobic core of the protein. Moving to next slide. We have also analyzed which beta sheets are prominent in stabilizing the upper form compared to the polar form. So it is found that you can see from the diagram that beta sheets 1, 3, 4, and 6 are of the upper form is more stable compared to that of the polar form. So the residues of these beta sheets in the hydrophobic core is stabilizing the protein structure. Moving to next slide. So coming to the conclusions, we have found that both apo and holoazurin are thermally very stable proteins. And also we observed a one-step unfolding transition between 400 Kelvin and 412 Kelvin. Also, upper assurine is found to be more stable than hollow form. Also, the one-to-one comparison of beta strands indicate that the 4 beta sheets of upper assurine is stabilizing it more compared to the hollow form. Moving to next slide, I thank my supervisor, Dr. Rajiv Vishwas, and also IIT Tirupati chemistry department faculty members for the help they have provided. I also thank my lab mate, Kamban Devendra Reddy, for his suggestions and help. Thank you all.
Yeah, thank you, dear Almin. Thank you so much. So next, may I request Dr. Moinak Ganguly. So he's from Madanapalli Institute of Technology and Science. So please, please come and present your work.
Sir. Yes, sir. Am I audible?
Yeah, you are audible. Please go on.
Thank you, sir. Okay, I am Dr. Ganguly and from MITS AP. Today my topic of talk is a nano trap of suitable mercury recycle technology with the formation of efficient ice nucleating particles. Next. Okay, so what is ice nucleation? Ice nucleation is a day-to-day phenomena from water to means liquid water to ice conversion. And ice nucleation can be homogeneous and heterogeneous. Mainly, heterogeneous ice nucleation is very common in our atmosphere or in our environment. And heterogeneous ice nucleation has different modes, say deposition, contact, immersion, and condensation. And most common mode is immersion mode, in which the ice nuclei is immersed in the fluid to form the ice. Okay. So here we are mainly going to discuss about the immersion ice nucleation, which is one mode of the heterogeneous ice nucleation. Next. Okay. So So in our laboratory, actually, we are able to synthesize the most efficient purely inorganic ice nuclei, that is magnemite, at the rate of mercurous chloride. We synthesized this. And we found that it is the most efficient inorganic ice nuclei. And its ice nucleation efficiency is very close to the most efficient but biological ice nuclei, Pseudomonas bacteria, known in the literature. Okay. And we also passivated our ice nuclei by clay minerals, say montmorillonite and kaolin, this type of clay mineral, to get the more surface area, which means to avoid the aggregation. And we found that it is much better also. So kaolin passivated mercurous chloride at the rate of magnemite. That is the most efficient so far known purely inorganic ice nuclei what we have synthesized in our laboratory. Okay. So next. Okay, and what is the mechanism behind the formation of such an efficient ice nuclei? To do this, we did lot of spectroscopic analysis, and finally we concluded that it is the lattice mismatch energy that is mainly the important driving force behind the formation of such an efficient ice nuclei. So this lattice mismatch energy means if there is low lattice mismatch energy, means the surface of the ice nuclei is more like the ice, then it will be a wonderful ice nuclei, and this happens in our case. We actually proved from spectroscopic ground. So this is the driving force. And so far known the most efficient ice nuclei, inorganic ice nuclei, is silver iodide, but it has some limitations. And our material is superior to silver iodide because it is sensitive to environmental conditions, environmentally available, cheap, and remain longer time in dispersed condition. Next. So not only it is a wonderful ice nucleating particles, but from this we are able to make a mercury nanotrap, because this material is wonderful mercury nanotrap. Not only it can remove the ionic mercury from water, but also it can remove the gaseous elemental mercury from the air. And recently it has been considered as VIP article in ChemSystem. So it can like control the mercury emission, and it can recycle it. And our technology is highly inexpensive, instantaneous, highly efficient, recycle, and energetically efficient, and totally green technology is that. Next. Not only mercury removal, but also our material are useful for the removal of the brilliant green dye. First, I just clarify that mercury removal is totally chemistry option, but here the brilliant green removal is totally physics option. And it can selectively remove the brilliant green dye also from the water, but it is physics option. And also with this we are able to recognize the nanoplastic present in the water using our material. So we can detect the brilliant green as well as nanoplastic in the aqueous medium using our material. Next. So I am highly thankful to my postdoctoral lab, McGill University, and different funding agencies, also IIT Tirupati, for giving me the platform to talk. And thank you, all of you.
Thank you, Dr. Ganguly. Thank you so much. So next I invite Ms. Ekta Nag from IISA Tirupati. So Ms. Ekta, please come.
Yeah, hello. Can you hear me?
Yes, please.
Hello everyone. I'm Ekta Nag from the IISA Tirupati. I will be presenting on my recent work based on the Trident Technical +2 catalyzed chemodivergent CH functionalization and the cyclopropionation of aromatic heterocycles. Next slide. Aromatic heterocycles, as we know this, aromatic heterocycles are everywhere in nature, and there is scaffolds found in many synthetic drug molecules that exhibit various biological activities. So especially like CH functionalized and cyclopropionated derivatives of heterocycle can be synthesized by using this metal carbon mediated chemistry. So here these are the previous reports where people have synthesized, and basically this kind of work is done by the use of late transition metal elements, which have several limitations such as high toxic metal waste and the high cost of heavy metal complexes. So these challenges can be overcome by the use of 3D metal complexes. Next slide. So encouraged by the previous reports and tried to overcome these limitations, we have synthesized the Trident base nickel +2 phosphine complex, which are acting as a robust catalyst. So this complex was synthesized by using the bulky SIB-based ligand treated with the nickel chloride in the presence of excess phosphine, resulted in the formation of very stable dark brown block-shaped crystals up to 85% yield. And this structure was further characterized by using this complex was further characterized by using the several spectroscopic and XRD techniques, where we have found that this complex is having the square planar structure with phosphine in the transposition. And we have also observed this complex is the key feature of this complex is the dissociation of the phosphine from the complex in solution. So which gives the which was confirmed by the phosphorus NMR, which is showing the two signals, two resonance, one at the 10.90 ppm corresponds to the coordinated phosphine, and the other at -5.30, which corresponds to the free phosphine. And the NB analysis also reveals that the nickel phosphorus bond is the electrostatic in nature, and it has a less bond dissociation energy. So 68 to 75% spontaneous dissociation of this phosphine from the complex, creating the coordinatively unsaturated active catalyst for carbon transfer reaction. Next slide. So we got the idea that this complex can be utilized as a robust catalyst. So we have initiated the catalytic activity of heteroaromatics in the carbon transfer reaction, especially in the field of CH functionalization and the cyclopropionation. And surprisingly, we have observed that the excellent chemodivergence with the different substitution at the end position of the heteroarenes. So its reactivity also depends on the electrophilicity of the carbonyl intermediate. So the presence of electron donating group at the end position affording the regioselective C3 functionalized product, and if the C3 is blocked with the other substituent, then the C2 is the major product. And in contrast, changing the directing group from the alkyl to ester electron withdrawing group under similar reactions conditions, we were getting the cyclopropionated product with the excellent exodia-stereoselectivity. Next slide. Then we come to the part where how this complex is showing the chemodivergence. So based on the experimental and theoretical investigations, we have proposed a mechanism where the very first step is the dissociation of phosphine from the complex and generating the monomeric active catalyst. This active monomeric species reacting with the carbon precursor, that is diazoester, and forming the carbonyl intermediate. And this And the NBO analysis also proved that the transformation of this intermediate is favorable as the nickel carbonate bond is kind of donor acceptor and have a high bond dissociation energy in compared to nickel phosphorus. And in the next step, this heteroarenes with the electron donating group at N position is reacting with this intermediate where the N lone pair of this nitrogen is participating in the delocalization and the forming the pi complex. And as the C3 is more nucleophilic and directly coordinating to the nickel center. And in the same time, the geometry optimization intermediate showing the elongation of this nickel carbonide bond, which confirms the weakening of this nickel carbene bond. And in the next step is the migratory insertion, where this carbene moiety is inserted into the inserted into the C3H bond and forming the C3 desired C3 functionalized product. So the last step is the dissociation of the weakly bonded expected C3 CH functionalized expected product from the nickel center and the regeneration of the active monomeric nickel complex for the next catalytic cycle. And if if we are using the heteroarenes having the electron withdrawing group where this carbonyl group is directly coordinating to the nickel center and C2 is attacking to the LUMO of the carbon and forming this six-membered organometacyclic, and this after the formation of this deuterionic intermediate, it gives the cyclopropanated as well as the C2 alkylated product as a minor product. So coming to the conclusion, we have reported the nickel plus 2 complex which can be used for the synthesis of biologically relevant molecules, and it is a very effective alternative to the moisture sensitive less abundant PCS metal catalyst. Next, thank you all for your kind attention.
Yeah, thank you, Ms. Ekta, for your nice presentation. Thank you so much. So next, may I invite Ms. Maria Francis from ISA Tirupati.
Hello, am I audible?
Yes, you are audible. Please go on.
Good evening, everyone. I'm Maria Francis from ISA Tirupati, working under Dr. Sudeep Roy. Today, I'll be giving a flash presentation on isolation of different oligomers of cyclic alkyl amino-supported alkali metal phosphinidides in solid state. Next slide. The reactive group 15 elements of carbenes are known as phosphinidides. They can be either synthesized in the presence of transition metals or in the presence or as carbene phosphinidine adducts. Metal phosphinidides are highly reactive species with low valent and low coordinate phosphorus atoms. We generally use to do different kinds of carbenes. One is N-heterocyclic carbenes abbreviated as NHC and cyclic alkyl aminocarbenes, which is abbreviated as CAC. NHC-supported phosphinidine was synthesized by the direct reduction of LP2L, where L is the NHC ligand, and whereas CAC-supported phosphinidines was synthesized by the deprotonation of CAC-PH by methyl lithium in THF. The previously reported synthetic schemes, they have in situ generated alkali stabilized CAC phosphinidines, but they haven't structurally isolated. Recent synthesis of Ruski group, they have tried to stabilize chlorophosphorins with the help of CAC. They have treated two equivalents of CAC with one equivalent of CAC PCl and generated CAC PCl in two different conformers. The cyclic voltammetric studies of CAC PCl shown the possible one electron quasi-reversible reduction at negative 2.85 volt. Next slide. Getting insight from the voltammetric studies, we try to use reducing agent potassium or potassium graphite to reduce cyclic CAC PCl. As expected, we could reduce CAC PCl and generate hexameric and dimeric forms of CAC supported potassium phosphinidinides. The spectral parameters of these structures are given in the table, and the HOMO and HOMO-1 are contributed by the lone pairs on the phosphorus. Next slide. Using the same synthetic scheme, we try to make the another CAC supported phosphinidine, where this time we have used dimethyl CAC. We again could generate, we could again structurally isolate the potassium phosphinidinide as a tetrameric unit. When we try to reduce the cyclic CAC PCl with sodium, the reaction didn't work. So we changed the reducing agent to sodium naphthalate and the reaction worked. So there we could isolate the trimeric unit of sodium phosphonadienide. The structural parameters of these two complexes are given in the table, and the HOMO and HOMO minus 1 are contributed by the lone pairs on the phosphorus. Next slide. This slide will explain the mechanism for the generation of CAC-supported phosphanidins in THF. One equivalence of potassium metal was reacted with cyclic CAC-PCl to produce CAC2P2, which was conformed by NMR. This indicated that the first electron transfer from potassium to CAC-PCl occurred to form the intermediate radical species CAC-P dot and KCl. In contrast, Na metal was not able to reduce CAC-PCl. The cyclic photogram of CAC-PCl show the first electron reduction corresponding to the one electron quasi reversible process, suggesting the generation of the radical. Once the high energy radical is formed, it can go to either two pathways, pathway A and pathway B. In pathway A, the electron transfer from potassium to the radical is crucial, whereas pathway B is highly probable dimerization of radical to produce a stable intermediate CAC2P2, which was found exothermic and more slightly favorable when compared to path A. This was evident from the fact that we could isolate CAC2P2 when the reaction temperatures were reduced. CAC2P2 underwent one electron reduction, forming CAC2P2 monoradical anion, which was further dissociated to produce the monomeric intermediate. In the reaction in THF-D8 show the formation of solvent-free dimeric potassium phosphonadienide and CAC2P2 evident from the 31P resonance at 186.3 and 55.9 ppm respectively after 12 hours, while the latter one was found to be the major product. In conclusion, we have reported the synthesis and the solid state characterization of CAC-supported oligomeric potassium and sodium naphthalate, sodium phosphonadienide by the reduction of CAC chlorophosphanidins using potassium graphite, and sodium naphthalate. Oligomerization of this class of complexes in the solid state has been found to be largely dependent on the charge accumulation of phosphorus atom, as evident by the significant calculated NPA charges on the phosphorus atom and the steric of bulk ligand CAC and the ionic gradients of alkali metal. Thank you.
Yeah, thank you, Miss Maria. Yeah, thank you so much. So we are left out with Mr. Kulbir. So let us see whether we can take him up. So Mr. Kulbir, so please.
Hello, sir. I'm audible now?
Yes, you are audible. Please.
Okay, thank you, sir. Good afternoon, everyone. Today my talk is titled as nitrite reductase reaction for the first time formation of hydrogen peroxide and a metal nitroxide. So please, next slide. So first of all, we should know why NO and why NO research. So nitric oxide is a gaseous molecule having one unpaired electron in its anti-bonding orbital. Because of its involvement in various biological processes, and nitric oxide nowadays nitric oxide become a very important molecule. However, excess of nitric oxide is a toxic molecule. All over the world, there is a lot of researches going on to understand the involvement of nitric oxide in biological processes. So in biological system, there are two sources of nitric oxide. One is nitrite reductase enzyme and one is NO synthase. So next slide. To mimic the nitrite reductase reaction, we have selected one cobalt nitrito complex, which we further treated with acidic media. On treating the cobalt nitrito complex with proton, we monitored the reaction with various spectroscopical techniques. In UV, it shows a conversion of nitrite to nitrosyl, emerging a new peak at 370 nanometer, which was proceed via an intermediate. However, this data is quoted by ESI mass and other spectroscopical data also. The final data is crystallized by XRD crystal data. To understand the mechanism of this reaction, we have observed this reaction using solution IR and EPR of frozen system. The EPR and solution IR is showing there was one formation of an intermediate which is further converting to metal nitrosyl. In solution IR, there was an information of a new peak at 1284 centimeter inverse. From literature survey, we come to know this peak belongs to a intermediate, commonly known as nitrous acid. And this peak was for cobalt O bound nitrous acid. From this, we proposed a mechanism from where this cobalt nitrous acid is undergoing NO homolytical cleavage and producing OH radical. And from OH radical coupling, we are getting H2O2. The H2O2 was trapped using proton NMR spectra, and the yield was calculating from the integration area of proton spectra. We got there is formation of around 55% of H2O2 in the reaction. So next slide, please. To confirm the radical mechanism, we proceed this reaction via 2,4-ditercibutyl phenol test, where we find the formation of dimer, catechol, and a nitrate product. From GCMS, we come to know the catechol product was the major product among these three, which can be explained by the formation of a phenolic radical, and which go under radical coupling to form a dimer, and which this radical same radical undergo with combination with OH radical to form catechol, and breakdown of this nitrous acid give a nitrite phenolic nitrite substituted product. So next slide. To conclude this work, we have demonstrated a nitrite reductase. We tried to mimic a nitrate reductase reactivity here, where we found that O-bound nitrous acid decomposed to OH radical and form H2O2, which may be the one of the pathways formation of peroxide in the biological system. And this also gave us a new pathway to activate the CH bonds of having low CH bond dissociation energies for the organic molecules. I would like to acknowledge ISER Tirupati and my supervisor, Dr. Pankaj Kumar Kohli, and my lab mates, and special thanks to IIT Tirupati for providing us a platform for presenting our work here. Thank you, sir. Thank you for your kind.
Thank you, Mr. Kulbir. Thank you. Thank you so much for your presentation. So now the session is open for discussions, questions, comments, and any suggestions to the presenters. So the session is open.
Don't keep more than 5 minutes, just 5 minutes.
I have a question for Devendra and Albin.
Okay.
Yeah, Devendra, you showed that number of hydrogen bonds in the first, second, and third shells to be same, and even in the bulk region same.
Yes, sir.
So what does that mean if it is same everywhere? Then how do the boundaries appear?
What, sir?
If the number of hydrogen bonds are same in each region, 3.5, 3.5, 3.5, 3.5, so how do you differentiate between the boundaries?
Sir, in second cell, third cell, 3.54, sir, not same like bulk water.
But 3.5, 3.5, they are all same. It's only different at the second decimal place. So number of hydrogen bonds at second decimal place probably doesn't matter. So that was my observation. I thought I will ask you.
Yeah, I think Devendra should later on discuss with his supervisor and then understand. Okay.
I have one more question for Albin.
Is the answer, sir. So below graph is the answer. It is average number is same, but the distribution is different.
Can I ask a question to Albin?
Yeah.
Yeah, Albin, you showed the azurine to be exceptionally thermally stable at 127 degrees centigrade unfolding.
Yes, sir.
Right, but the literature says that it unfolds at 83 degrees centigrade. And this is upper form and hollow form 83 and upper form at 70 degrees centigrade. So there is a difference of about a factor of 2 when you compare the upper form. So is there something different added in your studies?
Sir, in experimental studies, it is showing around 80 degrees Celsius. But when we are moving to computational studies,
There should not be
due to
that much difference. No, about 40-degree difference, 44-degree difference. So that's a very big difference.
Maybe you have to consider some more parameters in your theory. Optimization.
Okay, sir.
Thank you, Professor Nandakishore. So is there any other questions to the participant? I mean, to the presenters? So I have a question to Mr. Sai Manoj. Are you there?
Yes, sir.
IT Madras.
Yes,
So
sir.
I have a question. You talked about some theoretical analysis, right? So of
Yes.
organometallic complexes, right?
Yes,
Some
yes, sir.
cysteine isomerizations. So
Yes.
can you please tell what was the Hamiltonian? I mean, what is the exchange correlation functional you have used for the geometry optimization?
So first we have used it BP86 def to TZ-VPP level, and also checked with Minnesota functional MO6. So in both the cases, with and without dispersion, it is giving the cis is having the lowest, lower energy compared to the trans one.
So you mean even if you add the Fock exchange, still the C is predicted to be low energy?
Yes, yes.
Because BP86 is, you cannot trust. This is just a simple JGA functional.
Yeah, that's why I tried with other like Minnesota functional also to check whether it is an artifact, and also checked whether the Grimmy dispersion is also playing a role. So I tried like a different three, Minnesota with without dispersion and BP86 with and without dispersion. And in all the cases, like I observed, this is the lowest one. So in otherwise, like I am presenting the only BP86 level here.
Do you have any estimate if you vary the Fock exchange, how does the energy difference will come?
Yes, like in case of like if I change to like Minnesota functional.
The Minnesota functional is about more than 50% Fock exchange you are using.
Yes. So it is like around 7 kilocalorie it is coming, like when I use the MO6 level.
No, MO6 you have more than 50% Fock exchange. So if you use about 20 or 25% Fock exchange, did you try intermediate to look at what is the energy difference? Because 50% Fock exchange is too much for these kind of complexes. Do you have any estimate if you use like 15 to 25 percentage of Fock exchange in the Hamiltonian?
No, sir.
Okay. So you have the extreme 0 versus 50.
Yes, I mean yes, like so BP86. I actually like as I said, like I could not understand the this one the Fock exchange in the functionals properly.
BP86 is a pure GGA. You don't have Fock exchange, but pure GGA one cannot trust for these molecular systems. Okay.
Okay. Yes.
Okay. Yeah. Thank you. Thank you so much, Sai Manoj. And is there any other suggestions, comment for the presenters?
I just have one question to Ekta.
Yeah.
So,
Yeah, Dr. Gandhi.
Let us try to. That will be the last one. Okay.
Okay.
Yeah.
It's a very nice work. I just have a question. Since you proposed the mechanism for the P2CH oxidation of indole using this nickel carbene insertion to the P2CH bond, can we use the same strategy for oxidation of sp3 substrate? Did you check those substrates?
So, sir, actually we didn't try with the sp3 and all. We directly used this electron-rich heteroarenes. So we just focused towards this, like especially the C3 and other positions functionalization.
Okay, so Ekta, I have just a very simple question. It's not question. I'm curious to know. You have shown a lot of homolumo plots. No, what essential science we get out of it actually from your presentation? The homolumo distribution you have shown. No, so what essential science we are getting out of it?
So in the mechanism part.
Yeah, I mean, so wherever you have shown the homolumo distribution plot energy, no. So what science you are connecting with your molecule?
Sir, actually, if the like energy difference is like the next step will be the feasible if the like we have this low energy vacant orbitals. Like if it is we have the vacant orbital, then only like we can.
You are explaining the orbital interaction, right? I mean, in order to explain the mechanism, you are taking into account. Maybe I might have missed that one. I looked at the homolumo distribution, but I could not connect. Okay. But probably you have used that distribution to explain your mechanism. Huh?
Yes, sir.
Okay. Yeah. Thank you, Ekta. Thank you so much. So please join me in thanking all the flash presenters in these sessions. So please join me in thanking all. All of them have done excellent job. So now
Thank
I
you.
would like to just hand over to our Professor C.P. Rao, our HOD and convener of this symposium. So please.
Thank you. Thank you, Dr. Arun Manna, for conducting this flash presentation thing. Very nice. We have just come to the end of the program, and the concluding. All that I would like to say is we have cut across the lectures, other presentations across all areas of chemical science. And so if I have to put in a little, way that we can easily understand is that we had a really a feast with a lot of items, varieties being served, and but the serving plate is too small, and even after eating all that, we are still hungry. This is all I would like to put in terms of the concluding, and I would like to call upon at one or two from student side, a couple of faculty side to speak about the symposium without having any formalities. That means you don't need to thank and you don't need to address. Please kindly come up with any kind of observation or a kind of suggestion, both observation and suggestion kind of thing. I think that will be the best way that we can conclude the session. So couple of people from faculty side, couple of from the student side. Anybody speak? You don't need to address and thank, etc. Just say the problem, any kind of thing. Okay.
Sir, I would like to add a small
Yeah,
comments
please go ahead.
on
Go
the
ahead.
symposium. I'm Sai Manoj from IT
Great.
Madras.
Great.
As Professor Rangarao sir said that it is really challenging for us to present all the work in a very short time, but we try to make it. So I think it's a good exercise, and this kind of like usually presentations go for at least 15-20 minutes where we elaborately, detailedly explain. But to explain all the work, the main important points in such a short time is really a challenge. But it's I think we have learned a good it's a good opportunity for us to how to brief the results and explain in 5 minutes, like
Yeah,
I
yeah.
thank
No,
actually, yes.
yeah, yeah. So I understand. Let me address your thing. There are generally in conferences, there are three types: the invited lectures, young presenters, and then the flash presentation. So we didn't have the intermediate one because of the time problem. So we generally give for postdoctoral kind of level of people or young faculty somewhere in between, like 10 to 12 minutes or 15 minutes. Yes, we didn't have that session because of the constraint of one day. So totally we had now 8 hours of session, and we cannot have 8 hours of session in one day on online. So therefore we broke into two parts. Yes, if we can manage a 2 day symposium, all these suggestions can go into that. You're right.
Yes, sir.
So
But it's a good exercise for us also, like as a
yeah, sure, sure.
students to present.
Yeah,
Thank
yeah,
you.
yeah,
Yeah.
yeah, yeah. Now we have for young researchers there is 12 to 15 minutes usually used to have will be there. Yes. So but we could not have because of the short period. It's only one day. Great. Anybody else? It can be anything. You don't need it. Is Dr. Rangarao is there? Professor Rangarao?
Oh yeah, yes, yes.
Yeah, you want to say something?
Yeah, so basically
Other
this.
than other than what you told earlier, anything new?
Yeah, so anyway, this was actually planned in March, but it didn't happen. Maybe it didn't happen for good that we could meet online, and then this is a successful online uh, symposium. I would say first symposium in IIT Tirupati.
Yeah.
I think I must say that I think if all of you have put in good effort. I think usually in these symposiums, the things fail, the Zoom fails or something else fails. In fact, I was actually attending one similar symposium in the morning of third in ICT Mumbai. So they were using Google Meet and not Google Meet. time. I think yeah, Google Meet it failed, so they had to shift to Zoom.
Yeah,
There was some change, and
correct,
that did
correct.
happen. And I think all the lectures are excellent. I think our colleagues from Bombay, IIT Bombay, IAS Bangalore, as you said, it's a mix of many things, which is also important for the department which is just coming up now. Yeah, it's good to have a mix of everything, uh, so that they can actually decide which direction they can go. I think in future, one can have specialized themes in symposia. Uh, I think it's very important. The students are also learning now how to make presentations in online presentations. Like flash presentations is a good idea in my opinion. But generally, we give about 10 minutes, but that's okay. But this is a good practice for students also, because in future I am sure these kind of online presentations, online symposia are going to be norm of the day in future. So I think it's a good exercise. I mean, I must congratulate all the IIT Tirupati chemistry faculty and then speakers from other institutes, and also particularly the students who are very enthusiastic in giving their flash presentations. I must appreciate them. They were actually very good. All of them have done good job. That's what I want to say. I think we
Thank
should
you.
continue doing this.
Thank you, thank you. Yeah, yeah, yeah. Is usually practice 25 minutes, 12 minutes for the coming up researchers, and 4 to 5 minutes for the poster people. So that is what roughly the thing. The middle class we could not accommodate. That is 12 minutes one. So that's what I said already.
But
Yeah,
that's okay. I mean, it's
yeah, yeah.
well planned.
Future.
I would
Yeah.
say it's well planned.
Sure, sure, sure.
Okay.
And regarding the specialized symposia, we need a bit more time to get into that. Right now, we need to expand the faculty enough here. So we would like to have similar kind of things better, so that we can bring wider audience, wider speakers, a wider range of
But
speakers. That's very
but
good.
that will naturally will happen as the number of faculty increase
Yeah, yeah, sure.
in the department. Automatically, it will come. That idea
Thank you.
will come, and
Excellent.
they themselves will start opening up, and then they will say, okay, we'll have symposium on this topic, on that topic.
Sure,
That
sure,
naturally will come to the department over a period of time.
sure. Thanks a lot. Thanks a lot, Professor Rangarao. Uh, Professor Lahiri, say something.
Oh,
Other than no formality, just say
I,
whatever you feel.
I
Whatever you feel.
understand. I understand.
Yeah,
Well,
yeah.
it's a very good conference in the sense it tries to cover many areas in chemistry. As we all of you are aware of, that chemistry is passing through a difficult phase in the sense those who are senior, that it keeps changing so fast. And how to cope it up with this changing scenario, it's not very easy, because that chemistry, it has, as I said earlier also during my talk, it has the two components, right? Fundamental and its applications. And
Yeah.
application is very important in today's context. Either it is in the bio direction or in the industrial directions. Both are extremely important, of course, but not at the expense of fundamentals. So this balance is, that is very essential. If
Yeah,
the
yeah,
fundamentals are not strong, application cannot move, the way it desires to move. Right?
yeah,
So that balancing, and I am happy that during your this conference you tried to bridge between fundamentals and applications, materials, mostly bio or otherwise. So that's a good way to proceed on. The younger colleagues and particularly the students, they would know that what are the areas to be picked up for tomorrow and how to make a move accordingly. Now, I just noticed one point which you probably think about it for the future, that among the speakers, if I see the faculty speakers, there's no woman representation. Okay. Okay, now,
It's okay. Thank you very much. We will certainly. No, no, I fully agree. I know that there
what
is.
I see
Yes,
might be
yes, yes.
among the student slot, you have
Enough. We have enough
good
girls there.
number of girls over there.
Yeah, 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 yeah.
So that is always it gives a completeness of the
In,
system.
in fact, same thing. I had a thought last night when I was thinking about some of these things. But over a period of time, yes, we will be definitely covering many.
Yeah.
Yeah, yeah.
So,
Yes,
so
yes.
overall, I feel it's a very good conference. It covers many areas, and people are serious enough in talking, in discussing, and listening. Also, that is the most important thing, because we are probably serious in talking and discussing, but not listening. I have noticed whatever I could feel it through your emote that people are also listening very seriously. So it's of course it's because of the fact that you have invited a good number of quality speakers, including the students.
Yeah.
That's why it's a grand success. I should tell that way only. So wish you all the best.
Thank you. Is Dr. Prasad is there? KR Prasad. Rajiv, is he online? No. Okay. Professor Nandkishore. Professor Nandkishore, say something. No formality.
No, no formality.
Okay.
Professor Rangarao and Professor Lahiri has already covered most of the things.
Okay.
uh, and I want to also, express that this has been really a wonderful experience. And especially, when we are meeting online through the electronic platforms.
Yeah.
Yes, I have been, there for both the days, and there has been no disruption at all. So everything has gone very fine, very well.
Yeah.
And I have one suggestion that
Sure.
such meetings where you express that there is a wide variety of subjects covered which are connected to chemistry.
Yeah.
And as Gautam mentioned that the chemistry is changing very fast.
Yeah.
So obviously it will lead to several questions in the minds of youngsters. And there should be enough time for the questions to be asked.
Yeah. Sure.
So
That is very
somehow that goes, missed because sometimes we take a lot of time in finishing our lecture. So if there is a window for the students and others to ask questions, because that's what I also tell in my classroom, that if a question is asked by one person, then the 50 others sitting in the classroom, they get benefit of that question.
Yes, exactly.
So that is why I feel that this question answer becomes very important, whether it is taken up in the form of some additional 2-3 minutes given, or within that you finish your lectures early, or in the form of some sort of panel discussion later on at the end.
Agreed.
So that's the kind of suggestion I would like to make.
Agree. Panel discussion equivalent we are doing for flash presentation. For individual invited lectures generally immediately after the thing, but we can also have in future, yes. For example, you can see this particular thing. First lecture is 9:30 to 9:55, Professor G.K. Lahiri. 5 minutes there is nothing shown. 10 to 10:25 is K.R. Prasad. So that means we have built some the window period, but what happens is this window gets closed. The window gets closed because the wind outside is too much, so we are closing. So but we'll try. Yes, certainly I agree, there is a window. Yes, no, it's meant for that. Unless there are questions, it will not be interesting. It will not be interesting. Yeah, so we tried a bit more today. Yesterday we were a bit late, so we pushed faster. But today we gave more or less enough window, except after your lecture.
But overall, it has been like very great experience, and congratulations to you and your organizing team.
Yeah, this basically all these other five my younger colleagues deserve all that. I said they are senior because they are here before me, so that's why I address them as my senior colleague. But they are all young and energetic, so I hope they don't feel offended for that. So in that at IIT Tirupati, I'm junior to some of these people, and otherwise I'm senior. That's okay. So any other student would like to speak? Any other student, please come out. That will be the last thing. Student participation, other than the time parameter, yes, we notice time is given small. In fact, at IIT Bombay, we are two of the witness here. We were giving 3 minutes for all the students. I think you know that. Yeah, flash presentation. So yeah. Okay, so if there is no other query, question, yeah, Rajiv, do you want to take a photograph if everyone opens their videos?
Yeah, that can be taken, sir.
Is it possible? Is it possible?
Yeah, it is possible.
Yeah, can everyone please switch on your video?
Yeah, so with the sound.
Yeah, while you are doing that, let me tell you what I observed was yesterday beginning was we had 85 and we closed with 58. Today we have more or less 70 plus or minus 2 all through, except for just last few minutes because it's a hungry time. So otherwise we are 70 plus or minus 2 participants. So that was really very heartening. And I could see those in the these are all students in the course. They're there too. Okay. Yeah. How do you put them all? Is it possible in one
One
screen?
is not possible, sir. I'm taking couple of frames.
Yeah. Yeah. Yeah.
Combined. Yeah.
Yeah. Kindly switch on your videos.
So all of you, please switch on your video.
Yeah. GNVT, Sai, Vasu, Shivshankar, Prasad.
Jeez. Yeah. Venkat Krishna, Vashavi, Kiran.
Yeah. Yeah. So when you're clicking, just tell everyone will give you a smile.
Yeah. So you know. Okay, smile.
Yeah.
Okay. Many of them are not turned on. So okay.
Is it done?
You are doing it. Yeah, it's done. Sir.
Okay. Thank you very much. And now, Dr. Gandipan, please go ahead with the vote of thanks.
Okay. Yes.
Yeah. Thank you.
Yeah. Good afternoon, all. Already you probably tired, but just I will take 2-3 minutes and not too long because it's very important to thank each and everyone who is very important to make this event very successful. Okay. So first of all, on behalf of the Symposium Organizing Committee, I would like to humbly thank our Honorable Director, Professor K.N. Satyanarayana, for making it convenient to grace this occasion and delivering introductory remarks. Actually, I would like to promise to our director that we will ensure interdisciplinary speakers and participants in our future events as per his suggestion. I would like to extend our wholehearted gratitude to Honorable Director of ISET Tirupati, Professor K.N. Ganesh, for his esteemed presence in the symposium and enlightening us through his insightful opening address. Sir, we will ever remain thankful to you for your support. Of course, you extended always us to come and use your facility and have knowledge sharing with your department and the faculties. For that also, we say thanks here. Next, I extend my sincere thanks to our respected invited speakers, Professor D. Basavaya, Professor G. Rangarao, Professor K. Mangalsundar, Professor G. Mukesh, Professor Vijay Moganan Pillai, Professor G. Lagrin, Professor K.R. Prasad, Professor Nand Kishore, and our own Dr. Dabasis Mandal for their guiding and inspirational lecture on their expertise. In fact, we, the young faculties and our MS and PhD students are, some extent, we are unfortunate that we miss a chance to host you and have very close interactions with you at IIT Tirupati permanent campus. I hope this will happen soon in future, So we are expecting your love and the same support in future events as well from all of the invited speakers. Here I especially thank Professor G. Rangarao and Professor K. Mangal Sundar for their support to establishing this Department of Chemistry IIT Tirupati before we are all joining the institute. We had 25 wonderful class presentations from 11 institutions across the country. On behalf of the organizing committee, I congratulate all of you for your great efforts and success. I thank you all for your participation in this symposium. My sincere thanks also extended to all the listeners. I hope all of you would have got few take-home messages and research ideas. Thank you all. It's my time to thank my colleagues. It's slightly embarrassing, but I have to do that because they were working for a long time to organizing this whole event. In a chemical reaction, catalysts play a key role because it will initiate the reaction. Not only initiating the reaction, it will give you a very low energy pathway to overcome the activation energy barriers. Otherwise, the reaction won't take place. You know, our HOD Professor C.P. Rao is the catalyst for this symposium because he initiated this process and he suggested us the best pathway to organize this event. Without his effort and without his effort and the initiative activities, this symposium could not be possible at this great success. So thank you, Professor Rao, for supporting and guiding us. Next, my sincere thanks goes to Dr. Rajiv Biswas, co-convener of this symposium, for organizing all works of this event. I extend my gratitude to my colleagues. Dr. G.P. Roy is sitting close to me, and Dr. Arunamana next to him, and Dr. Tabasis is right hand side to me. So all of them were working a lot for this work, for the symposium to make it a great success. So I thank you all for your effort. Next, my heartfelt thanks goes to our departmental staffs, Dr. M. Jagdish, Dr. G. Sanyasi Naidu, and Mr. Ajmira Nagu. They were always available to help us in all process without any time limits. Anytime we can call them, ask for help. It's no time, it's 10 o'clock or 11 o'clock at night. It's no matter, they will be there with us to help us in all the local processes. And I also thank all the members of our institute from administration, academic, IT section, and all other departmental colleagues for their constant support. Of course, last but not least, our students, the motive of this symposium is to facilitate our students to interact with or to getting closer with the great minds of chemists all over India, and that is the main motive for our department. That's the reason we are organizing these type of symposiums, so that our students actively participated in this symposium. So I also thank them. Here they have limited participations in action because they are all in home. I hope in next sessions we will ask them to participate in action as well instead of listening alone. So yeah, with that, I thank you all again. And thank you very much.
The only one person who has not been thanked is yourself. So let me thank you in that case. Okay. Okay. Thanks a lot to everyone, and we will see in some other occasions again. Okay. Bye bye. Is it okay?
Yes, sir.
Yeah. One thing is we will be sending a link for you to download the brochure, the program abstract brochure. Okay. So kindly send that.
Yeah. Thank you, sir.
Great. Okay. Bye bye.
Okay. Bye bye.
Thank you, sir. Everyone, bye. Everyone, bye. Yeah.